We begin the first presentation in 2 or 3 minutes. Once again, please feel free to use the chat to exchange already, but please don't unmute yourself except if you are a speaker. Okay. I think all technical things are okay. And okay, we are already 50 people at the moment. Virginie, no answer from Brian, I suppose?
Not yet.
Not yet, okay. Okay. So, Irina, I saw that you are here. So if for some reason, probably because of the difference of time, Brian is not there when we start, we will start with you because you are, if I don't make a mistake, the second person to speak.
Yeah. And hello, everybody, by the way. Hello, Frank. I see many.
Hello. Yeah.
Good morning.
Good morning, evening, depending of the time, the place, and so on. Okay. So, yeah. It's yeah, Greg, you are there also. So there is only Brian who is not here. Okay. For the first speakers. Brian is just coming. Great. Okay. Good. So, because this will be a very long conference online, well, 6 hours, I propose to start at almost on time. So welcome everybody. This is the 5th Eurosymposium on healthy aging. It's the first one only lasting for 1 day. It's also the first one only online, and it's also the first one with probably more than 100 people who will join at one moment or another. Please feel free to give your opinion on the chat. Thank you everybody for being there. Thank you especially for the speakers, but also for all participants. And thank you for the people who were making this possible, among other people, especially Ilya Stambler and Virginie Stephen, who were working, I mean, almost as much as I was working, maybe even more sometimes. Today is the 1st of October. The 1st of October is the International Day of Older People. It's also for us the day of longevity. And today Virginie will put the link to the text that we hope to adopt at the end of this meeting concerning biomarkers and clinical trials. You are welcome until the end of the meeting to make comments, to suggest modifications. So this is all for my introduction, and now I give the word to Sven. Don't forget, okay. Sven. Yeah.
Well, I want to say welcome to everyone as well. So due to Corona, we had to postpone, as Didier already explained, the main conference to next year. And we hope to see you there, of course. So today we will have a little bit of a special edition of EHA, one that is devoted to more or less a single main topic. But it's a very important topic, and that is the question of how can we translate the multitude of basic research that is going on to benefits of the patient. Or if we use the famous slogan of the American cardiologist Spencer King from 1996, from bench to bedside. In the first part of this meeting, we will discuss clinical trials on aging, and we will undoubtedly learn about the ongoing trials, such as TAME, and also learn about how these are structured and what changes or improvements or suggestions there are for future clinical trials. Next, we will discuss the development of biomarkers on aging that can be used as surrogate endpoints for human clinical trials. Conducting trials with mortality or even the incidence of age-related diseases as endpoints, starting in healthy elderly patients, is cumbersome and very expensive due to the long time frames needed to establish efficacy of the interventions using these endpoints. Therefore, the development of validated surrogate endpoints of aging will be a potential solution to this conundrum. Finally, we will look at the legal and the socio-political aspects of translational research in humans on aging and what can be done to stimulate the establishment of future human clinical trials on aging. To conclude the meeting, we will have a general discussion followed by the formal adoption of a declaration on translational research on aging. So questions can be asked in the chat, and if time permits, we will ask one or two of these questions to the speaker. We kindly ask speakers to answer any questions that are in the chat after their lecture is done. And so due to time constraints, please stick to the allotted time for your lecture. Thank you.
Thank you, and I also must say how happy and proud I am to be in such a distinguished audience of speakers, of attendance. A very important meeting on the key issues of longevity research, the trials and biomarkers that makes it possible. Just a couple of technical questions, as Sven also said. If you are not speaking, please mute yourself. If you are speaking, please stick to the time. There are many speakers, so please aim to speak about 10, 11, 12 minutes. We will make signs when it is close to the time, and there may be a question or two in the end. And another issue I wanted to mention is that, as Didier also mentioned, this is a part of a larger campaign, of a Longevity Month campaign, starting with Longevity Day, the 1st of October. This campaign we do every year for about 7 years. Hundreds of events, promotions, meetings, publications are done by activists. So you're also welcome to organize more events, more meetings, more promotions during this month. And there is even a prize for this from the International Longevity Alliance for the best longevity event or promotion during the Longevity Months, $3,000 first prize, $1,000 second prize. Well, let's make it a real event to boost the awareness about longevity.
Okay. So for the first part, Sven will be chairing. So we are beginning just a few minutes too early. That's great. So we will have more time for questions and remarks. Sven, you can share now. Unmute yourself.
Yes, so the first speaker is going to be Brian Kennedy. So I give the word to Brian.
Well, thank you. Thanks for inviting me. And I have to say that I managed to come down with a stomach virus or food poisoning or something. So I'm going to try to hang in there through my talk, but I may not make it much beyond that. Can you see my slides?
Not
No.
yet. Not yet.
Hold on. I think I forgot to share.
Oh.
It would help to share.
Yes.
How about now?
Yes.
Yes.
Thanks. And so I'm going to take you through a little bit about some of the research we're doing, and also how we're thinking about doing clinical studies here in Singapore, particularly once we get up and running, which will be in the next month or two. the virus is more or less under control here now, but it slowed down the research quite a bit for a while. This is a slide that talks about interventions for aging, and I thought I would start here. I stole this from Centara, which is a company thinking about how to tackle the aging process. And I think it nicely illustrates that there are three types of interventions that we're all thinking about. And this inner circle here in dark red is really lifestyle interventions, which are things that I think we know pretty much work already. I think that, for instance, exercise, some combination of cardiovascular exercise with resistance training, I think is very, very likely to extend median life expectancy and extend health span. I think we can debate about whether it would affect maximum lifespan. Diet, I also think stress management and sleep quality are things that could be manipulated here, among others. A lot of work has been gone into understanding how these lifestyle changes might affect aging, but the challenge is really getting compliance. So even if we know what to tell people to do, they don't want to really do lifestyle change in a sustainable fashion. And so the question here is really, how do you get widespread adoption of any method that you are trying to invoke. The second circle here is really, I think, where we are now, which is drugs and small molecules, natural products that might impact the aging process. And we're working heavily in this area, as are a number of investigators that will be speaking tonight. Then I think there's this third circle, which is more radical change. I would put gene therapy, stem cell approaches in there, although maybe they're starting to move into the second circle, I suppose. But these are things that may have a much bigger effect on aging, but are really not ready for human studies to a large extent at this point. So we're working on a lot of small molecule interventions, as I was talking about, and these are ones that we're playing with in the lab right now, mostly in preclinical models. With several of these, we want to study in human clinical testing as well. And I'll tell you how we're going to go about doing that. I wanted to just show you some data on alpha-ketoglutarate because this is a paper that got recently published. It's a natural product that's extremely safe, and it's something that we're excited about testing in humans. This was sponsored by a company, PDL Health. I won't spend much time talking about it, but alpha-ketoglutarate has really a modest effect on survival. So if you started at 18 months of age, it extends lifespan in females by up to 10%. In males, it's a smaller effect. In any one cohort we've done, we don't see significant lifespan extension in males, but we also always see almost the same thing, which is a slight extension. And if you combine the cohorts, you would get statistical significance. We have 4 cohorts now, which show pretty much the same thing, of black 6 mice. They show pretty much the same thing with lifespan extension in females and only a small effect in males. So this is not really why you might get super excited about this. It's an effect, but not a huge one. I should point out before I go on that this was done in large part by Daniel and Azar in my lab, as well as Gordon Lithgow's lab at the Bach. The reason we got excited by this is that it has a much more dramatic effect on frailty. So we think that AKG in mice is compressing morbidity. So if you look here on the left, these are individual frailty scores per mice using Susan Howlett's 31-point frailty index that was developed for mice and that corresponds to some extent with human frailty. What you see is that the blue dots go up faster than the pink dots, indicating that the control animals are getting more frail than the AKG treated animals. But this is a complicated way of looking at this data. This is every dot is a mouse taken at a different month of age. But some of the mice are dying throughout the course of this experiment. For instance, this control animal here at 23 months with very high frailty was actually dead by 25 months, so it drops off the curve. So another way of presenting this data is to normalize it based on the lifespan of the animal. So all of these dots, for instance, are taken between 50 and 60% of the lifespan of the animals, between 60 and 70, 70 and 80, etc. And if you look at it that way, you get a much more coherent observation that frailty goes up pretty steadily in the control animals, but in the AKG treated animals, it's largely flat. In fact, we get a 50% reduction in frailty as measured by area under the curve. This is true in both females and males. And so the upshot is there's a much more robust effect on frailty than there is on lifespan. So what's alpha-ketoglutarate doing? Well, I think it's like a number of small molecules that we've linked to aging. NAD would be a good example. It's kind of hard to understand mechanistically what it might be doing to affect the aging process. Because alpha-ketogglutarate's in about 1,000 chemical reactions. It's also a central metabolite in the citric acid cycle. So narrowing it down to one or two activities is a bit of a challenge. We've shown that it improves adult stem cell function, improves metabolic flexibility, enhances respiration. There are interesting effects on microbiome we don't understand yet. It reduces inflammation, and in human anecdotal data, at least, we see effects on epigenetic clock. But I want to talk about one activity which is very simple of alpha-ketoglutarate, which may be affecting aging. It's something that we tend to overlook. And that's the effect of AKG on the glutathione production. So if you look up AKG on the internet, you'll find several articles. These are just 3 that I pulled down, showing that alpha-ketoglutarate can enhance athletic performance, bodybuilding, healing from injury and wounds, which is a major point of muscle strengthening. And so bodybuilders and exercise people have taken AKG for quite some time. And we were wondering why. Some people think it's because of anabolic activities, but I think there's another activity that may be relevant, and that's glutathione production. So it turns out that red blood cells with deficient mitochondria need to make things like glutathione through different mechanisms. And so unlike most cells, red blood cells can directly take up alpha-ketoglutarate, where it goes in to produce glutamine and then glutathione. And in fact, tracing studies have shown that 89% of the glutamate comes from alpha-ketoglutarate in red blood cells. And of course, red blood cells need to have a lot of antioxidant defense mechanisms because they're carrying lots of oxygen around to deliver to peripheral tissues. And so what we've been able to see is if you add AKG to red blood cells, you get a pretty robust increase in alpha-ketoglutarate either in vitro or in vivo. These are mice treated for a week with alpha-ketoglutarate, and you can see increases in levels. And so that leads us to a pretty simple model that we're trying to test now. One is that glutathione, well, so just to summarize that part, glutathione decreases with age in red blood cells. Unlike many cells, red blood cells readily take up AKG for glutathione production. Their red blood cells are also particularly sensitive to the lack of glutathione. So we think that restoring glutathione levels in these cells probably underlies some of the exercise findings, but also perhaps with aging improves oxygen delivery to muscle. And that may be one component that affects the aging process. And so there are 2 clinical studies ongoing now with AKG. One is sponsored by the company, and that the product that from the company actually has vitamins in it too. The male product has low-dose vitamin A, and the female product has low-dose vitamin D. But this reflects, both of these studies reflect how we're thinking about doing aging clinical approaches. So we're looking at people 45 to 65, relatively small trials, placebo-controlled, 9-month interventions, and then looking at aging biomarkers and inflammatory markers, in this case particularly as outcomes, because we saw big effects on big anti-inflammatory effects with AKG in the animals. And then we're doing another study at NUS that will be started hopefully in the next few months, which is similar, except that we're going to only be looking at the alpha ketoglutarate at 2 different doses because we want to understand more about mechanism from this study, but looking at similar parameters as well as a number of physiologic parameters as well. And I think this brings me to how we're thinking about outcomes for clinical trials. You know, there's kind of 3 approaches that have been taken. A number of companies have taken the approach of trying to take longevity interventions and then trying to treat disease with that. Now that makes sense if you want to get FDA approval and reimbursement because it's maybe the easiest route to do that. But it's also kind of a sleight of hand. You know, you're developing something that's really having a preventative effect on aging, and then you're trying to find a disease that you can therapeutically treat and to get some sort of outcome. And I feel like that, especially given some recent high-profile failures, I feel like that we should, if we're going to try to study aging, we should try to measure aging as an outcome. And I know that this may be a better route to get approval and reimbursement and make money, but I also think that it's not directly looking at what we really care about, which is the aging process. So that leads to 2 other kinds of studies. The preventative approach, where you're really trying to look at health span directly and using aging biomarkers. And you all have heard of the TAME trial, of course, you'll hear more about it later. And I think this is a great study with metformin, the idea to prevent multiple diseases simultaneously to show that you're actually impacting human health span. So I'm very supportive of this approach. It's also a very expensive approach, however, and the trials are long. And so in Singapore, that's really not amenable to us. And so we're taking a different approach using these aging biomarkers. And we think this is really amenable to lifestyle interventions and natural products, particularly, although we will test drugs in this context as well. And I would admit that some of these modern biomarkers, or while they're not fully validated, I think the data on them is quite impressive. And so I've become a believer in using them to measure aspects of biologic aging. So one of the things we're trying to do is to align studies between humans and mice, so that instead of looking at survival in mice now, we've switched over to doing 6-month interventions starting at 18 months of age and using frailty, as I've shown you, and also biologic aging markers as outcomes, because these are more similar to the clinical studies we'll be doing in humans. So the idea is that we can take several interventions in a relatively agnostic way and work back and forth between the clinical studies and the animal studies, trying to align them as much as possible and trying to learn how correlated these 2 organisms are in the context of mitigating aging. And then we're also adding mechanistic approaches. In some cases, we do look at disease models. And we're working hard with scientist Dean Ho here at NUS to look at precision dosing, so that we're now doing studies where we might give 30 mice a small molecule like AKG, but we're measuring blood markers along the way and then trying to change the dose in each individual animal to optimize the outcome. And so it's not clear that this is going to work or not yet, but if it does, we'll try to move these studies into human clinical studies as well, the precision approach, I mean. And so we have a range of things we're measuring. This group in the middle are really biomarkers of age, DNA methylation you know about. We're using Jackie Han's system in China to generate biologic age from facial reconstruction. We're generating age from complete blood counts for using some data from Gero. We're also using metabolomics data and a few other measures like this, partly to see how these different biologic markers correspond to each other in Asian populations. And then we're combining that with more standard measures like arterial stiffness, inflammation, and a few other things as well. And hopefully this will allow us to generate a biologic aging signature in Singapore and use these measures, either individual measures or an integrated measure, to determine which interventions are working the best. And so, as I said, it's taken a while to get these studies started. We would have been underway already for a while if it hadn't been for COVID, but we're pretty much there now, so we should be started in the very near future. And so in Singapore, I just show one slide. This is highly relevant, as 30% of the population is going to be over the age of 65. The childbirth rate in the Chinese population is 1.1 per couple. And so the aging demographics are changing rapidly. And so this is really one of the ground zero places for aging. And so if we can extend health span, I think all of you probably get this. And probably the most important thing is we're going to dramatically improve life quality and independence late in life. But we can also extend work years. Singapore, like many countries, is raising retirement age. We can keep the government from having to build hospitals. And in fact, we've estimated that we can save the government $3 billion just by extending health span 3 to 5 years in Singapore. And then finally, what we'd like to do is bring in sort of a biotech longevity industry into Singapore. And we're in the process of having some impact on that, bringing companies in. And we're actively looking to support longevity companies in this area. The government is very supportive with taxes and other forms of non-dilutive financing to bring biotech companies in. So we're working with them on that. So with that, I think I'll just stop and take questions if you have any. Thanks a lot for your time. These are all the people in my lab and the collaborators that helped out on the project. And thanks again.
Okay, thank you for your lecture. So there are quite a few questions that came in. Jos asks, how much alpha-ketoglutarate is normally in the human body before supplementation?
That's a good question. I mean, and one thing we've noticed is that if you give animals alpha-ketoglutarate, it doesn't persist for a very long time. So it's not like a drug, I think, in that you need to have it around to inhibit some enzyme. We think we're actually feeding chemical reactions by adding alpha-ketoglutarate. So it's like a little bit like NAD. You add a precursor to NAD, it goes up and then goes down really rapidly. And it may be the reactions it drives that are really having the biggest effect. So I can't give you an actual quote on how much AKG. There's a lot, of course, because it's a required component of the TCA cycle. It does go down with age, and we can see supplementation when we add labeled AKG. I think what got us confused trying to look at mechanism was that the labeled AKG we add doesn't go into a lot of different cell types, and that comes back to a point I alluded to, which is that many cells don't take up AKG directly, and that's why we started focusing a little bit on red blood cells. But it also may be that AKG gets converted to glutamine, and that gets converted back to AKG or other things. So we're still trying to sort that out.
Okay. A question related to that actually is Nirbu asks, what happens to white blood cells?
What happens to what?
Yeah, when you give alpha-ketoglutarate. So is it taken up or?
What kind of cells though? I missed your
white blood cells.
Hold on, let me look at the question. I'm still not understanding you.
So the question is, what happens
white
to
blood cells? Sorry. I'm trying to hang in there tonight. We haven't looked directly at other white blood cells and other types of lymphocytes in the blood. It's not. I don't think that there are active mechanisms to take that up, but it's something we're going back and doing in more detail now, because in some of our initial experiments we weren't paying attention to blood cells at all. So, like for instance, we looked at muscle. We looked at intestinal cells and muscle cells where there is some AKG taken up when we give it to the animals. But things like muscle and fat, we didn't see a lot of AKG uptake. But we're now going back and looking at a wider range of cell types.
Okay. I think we have to take one more question and then we have to move on. So John asks, how long is the half-life of alpha-ketoglutarate in the blood? And so is it better to take it in small doses throughout the day or one big dose once a day?
It's relatively short. The way we give it to the mice is in their food, and so they're eating it all day long, and we think that's important. And actually, the company produces alpha-ketoglutarate in a sustained release format, so it is released more slowly in the intestine based on its formulation in the pill form. I can't. We haven't done side-by-side comparisons, but we think that may be important to seeing the biologic activity. Also, I would point out that a lot of the AKG you buy on the market is bound to arginine or other amino acid analogs, and we were hesitant to add amino acids like arginine because we didn't know what the effects on aging might be. So we haven't done a side-by-side comparison with calcium alpha ketoglutarate versus arginine alpha ketoglutarate, however. But I suspect that some kind of sustained release or lowered, taking it over a longer time frame would be beneficial.
Okay. Yeah, I think we have to leave it here and go to the next speaker. Thank you so much for your lecture. And if you feel able to, you can answer some more of the questions in the chat. Okay,
I'll try to answer a few. Thanks.
So our next speaker is Irinia. Can you wait? Hello, Irinia?
Yes, Irina is already sharing the screen, so
yeah,
it's great.
Sorry, sorry guys. Yes, I shared my slides, and I will start the video. Yep. Now you guys can see me and hear me as well, right? Yes, so I can go ahead and present my talk.
Yes, we are set.
Okay. All right, thank you so much for inviting me, and I'm very happy to follow up Brian's presentation. Brian and I know each other, in fact, for many years. I think the first time we met, he was associate professor, and he invited me to give a talk. So that puts things in perspective. So I usually start my presentations with a slide which shows that yes, organ deterioration and disease are increasing exponentially with advancing age. However, I compare rat and squirrel. At the same chronological age of 3 years, rat will be old and full of disease, and squirrel will be quite young and healthy. And so though even these animals are of the same size, relatively speaking, and squirrel even has higher metabolism than rat, they age differently. And the bottom line of this, or conclusion, is that the rate of aging is unlikely to be controlled by the accumulation of the damage, because these animals lived on the same planet, at the same entropic environment. But from our perspective, the rate of aging is controlled by the degree of repair, how much we can repair the damage. And here I would like also to introduce in my lecture the point that if you are proposing a certain treatment that we believe is anti-aging, it would be better to understand why that treatment might work. For example, rapamycin, why inhibiting production of ribosomes and cell function more generally, protein production and so forth, why that would be rejuvenated. Or metformin, why would inhibiting mitochondrial activity and production of ATP be rejuvenated? So in other words, metaphorically speaking, if you would like to throw a rock, it doesn't matter whether you do or do not know the law of gravity. You will throw the rock successfully at its target. But if you would like to launch a satellite on an orbit and that satellite has a lot of transistors in it, bunch of transistors, you cannot do it without in-depth understanding of physics. And so then our general approach is that we are not trying various substances. We try to understand what controls the fundamental process of aging, and through that understanding, develop therapeutics. And so the first point that I'm introducing, let me remove this one here, is that life and health span is controlled by the efficiency of repair or regeneration more so than by the accumulation of damage. And this understanding came from kind of comparison between young and old person. So young and old persons will experience relatively the same damage, but young person has better repair. And then what would happen if we induce experimentally and clinically better repair capacity in old person? Would that mean that the old person now is younger? So that is kind of in a pinch our experimental hypothesis and approach. I will briefly introduce our experimental systems. We look at rejuvenation. What Brian showed you is the delay of health decline. Can mice become healthier even later in life? But we look at robust reversal of aging. Can we start with an old mouse or old person and make them significantly younger and very quickly so? And the idea there is that for everything that we understand, for example, antibiotics work, and if somebody is infected, then we cure them. They become non-infected. They do not have bacteria. So can we then extrapolate this to aging? If somebody is old, can we cure them? Could we make them young if we truly understand the process? So we are interested in robust changes from old to young. We look at mesoderm rejuvenation, for example, at muscle, where we reduce fibrosis, inflammation, and improve muscle repair. And the derm rejuvenation, where our experimental model is liver. And there we look at the reduction of fibrosis and adiposity and improved regeneration. And ectoderm rejuvenation, when we look at the brain. Can we make brain form new neurons better, reduce neuroinflammation, and improve animal cognition? Our axiom is that when several key organs become significantly younger and healthier, we can really say that the whole animal is younger and healthier. And the opposite, in fact, is not true. I'm very glad that Brian Kennedy was demonstrating to you the extension of health span and not just lifespan, because in lifespan studies, very sick old mouse could be just sitting in the corner of the cage and shivering, but it would be still considered in the positive column. It is not yet dead. And the fact that we notice, which might be of interest to this audience, is that younger, healthier tissues are impossible to coordinate without good metabolome, good organelles, good proteostasis, exosomes that are healthy, gene expression, and epigenetics. So once we look at the tissue health, we simultaneously know that multiple attributes of that health are also in place. The first step in the mechanistic understanding of aging and anti-aging was, is aging malleable? For example, once we become old, can we become much younger? And we approached it in hydrochronic parabiosis studies, which are shown here in comical way. And then later on in much better defined hydrochronic blood exchange or apheresis studies. And I would like to know that this specific setup and experimental system was really encouraged in our laboratory by Aubrey de Grey, who first just encouraged and then insisted that we do it, and then provided funding, and it turns out to be an excellent experimental system. So in both of the systems, multiple organs and tissues become rejuvenated. You start with an animal that is analogous to 75, 80-year-old person, and you make that animal analogous to about 30-year-old person. Very quickly, in a matter of weeks in parabiosis, and after one single exchange in hydrochronic apheresis. So that tells us important things mechanistically, that regardless of telomere attrition, telomerase activity, epigenetic status, changes in metabolome, all of this is transient and could be quickly reset to younger states. So aging is reversible. Additional point here is that hydrochronic apheresis in small animals and mice is a miniaturization of FDA-approved procedure for people. So from that, there was highlights and some cartoons about young blood being the rejuvenative secret sauce. But even though in parabiosis and blood exchange, many things are similar, not all of them are similar, and that gives us additional intuition or additional insights about what aging is and how to reverse it. For example, in parabiosis, where mice share not only blood, but also organs and environment is changed, formation of new neurons in the hippocampus, shown here as green dots or red dots, and hippocampus is the area that is important for learning and memory. So formation of these neurons declines with age dramatically, and it can be induced, improved in old animals that share blood with young animals, which we discovered first back in 2004. And after sharing this data with Tony Weiss-Corey, his laboratory embarked on similar studies and repeated what we found. And then young mice suffer from being sutured to old partners. So their neurogenesis declines in hippocampus. So that happens when animals actually physically suture to each other and they share many other things, not just blood. But if you look at what happens in just blood exchange, again, young mice exchanged with young blood have better neurogenesis than old control animals. And this neurogenesis declines through one single exposure of young animal to old blood. It is obliterated. So old blood is really inhibitory. But there is no improvement in young mice after 50% of their old blood was replaced with young, putting a big question mark on whether young blood is a medicine. And there are additional things that I do not have time to show. And this is an example of functional test, which is called four-limb hanging. Mice have to hang on an upside down inverted mesh, and they learn how to hang. So in the first trial period, young mice try to hang, and then old, and then eventually they drop from the mesh to the soft bedding. And it is not a brute force test, so there is no statistical significant difference between young animals and old animals. They actually like doing that. They usually climb on the tops of the cages and hang as much as they can. But then after this test or trial or exercise, when you retest them again, if young mice were exchanged with young blood, 3 out of 4 now learn how to hang, and they display better agility and cognition, memory. You need to have many different parameters to learn how to navigate this test. But if young mice are exchanged with old blood, 3 out of 4 now are not statistically different from the control cohort, so their cognition was poor. And once again, old animals, regardless whether they have old blood or 50% young blood, are not better. So that tells you something, right? So that is the science from which we then make the next step. The next step for us was that the intuitive jump to young blood being the medicine was inaccurate. And then additional, there was a lot of data for that in our laboratory, which we were running at UC Berkeley with Dr. Mike Conboy and myself for over 10 years, 15 years in fact. But among this data, one thing that you might be interested to know is that if you mix young blood and old blood and expose cultured cells to that mixture, old blood dominates and inhibits performance of even young stem cells. Again, indicating that young blood is not a medicine and old blood is a problem. And so that brings me to the paper that we published recently in Aging. By the way, it was reviewed in Nature, and then after extensive reviewing, we were suggested to do yet more experiments and reply to reviewers' comments. And I'm not a very patient person, so instead of doing all of that, we decided to publish this study elsewhere. What the study demonstrates is that young blood is not a medicine, and furthermore, surprisingly, it is not needed for rejuvenation. In other words, the fact rejuvenation takes place without a putative cause, young blood. And instead, old plasma dilution is robustly rejuvenative, and it is, in fact, more rejuvenative than many other current approaches on silver bullets or so forth. So here we modified the procedure of isochronic exchange in small animals by exchanging young or old mice with saline and albumin and returning their cells back to the animal. So the cells remained the same age as the mouse, but 50% of their plasma was diluted with physiologic solution that has no age. So we call it neutral blood exchange. As I mentioned, the same procedure is approved by FDA in people. Here's some somebody's noises, so if you guys can maybe mute your microphone. You guys hear the squeaking? I don't think I can continue. Oh, thank you. Right. So here we tested a couple of things. One is that if you have an old animal and there is no young blood, you simply dilute all blood factors by 50%, will it become older? Because if you postulate that young blood is needed for youth, then young mouse, when you dilute young factors, would become old. And in the opposite end of the spectrum, if you have old mouse and you dilute 50% of its blood serum, will it become younger or not? And so the answer is shown here for muscle. And specifically, you are looking at the newly regenerated tissues where white areas represent the bad outcome of fibrosis, and these blue dots and white areas are bad. And then these newly formed muscle fibers represent a good outcome. And you can also look at it through immunofluorescence for the marker of the newly formed muscle fibers that replace injury site, which is called embryonic Meissner heavy chain. And so by all of these metrics, either looking at regeneration, fibrosis, or how healthy the new muscle fibers are, single procedure of blood dilution, plasma dilution, rejuvenated old animals to the degree that they are not different from young. That is really the only outcome, to my knowledge, where old animals now become indistinguishable from young, at least in this metric. And the same will be true for reduction of fibrosis, and the same will be true for the improving the health of the newly formed muscle fibers. And in all of these experiments, young animals did not become older when we diluted their blood plasma by 50%. They are still young. And because this procedure is FDA approved, we had a collaboration, and this collaboration is ongoing right now. In fact, as phase 2 clinical trials, collaboration is with Professor Dobry Kiprov, where we analyze what happens to humans that underwent this procedure. And we also analyze the effects specifically of albumin, human serum albumin. Is it important, or is dilution more important? And what I'm showing you here is that if you look at the proliferation of muscle precursor cells that are exposed to serum, blood serum from old individuals, before procedure, their proliferation is low, but after therapeutic plasma exchange, one single procedure, their responses are high. So instead of being inhibitory, their blood plasma now is supportive of regeneration. And the same will be true whether or not you add albumin, and by many reasons, we do not think that albumin plays a major role in the outcomes of our experiments. As I mentioned, we also look at brain and liver, and in the brain, we focus on that area of hippocampus called subgranular zone, where new neurons are formed from the dividing neural stem cells. And once again, remarkably, one procedure of mutual blood exchange, which we tested in many experimental animals and independent experiments, made all neurogenesis indistinguishable, statistically similar to that of the young neurogenesis. There's not 20% improvement, it is really the same. And then diluting plasma of young mice did not make them older, although we have a big spread in their neurogenic responses. And the same happened in the liver. We look at liver adiposity, which is increased with age and is reduced by neutral blood exchange, and liver fibrosis, which is increased with age and is reduced by neutral blood exchange. Once again, the key take-home message is that single procedure of neutral blood exchange is robustly and rapidly rejuvenative, as one would expect if you actually uncovered the mechanistic cause of aging and therefore uncovered proverbially so-called antibiotic against aging. You will see effects in multiple organs and tissues, and the effect would be that of a cure. And then we looked at proteomes of animals that,
excuse me, but we are running late, so I think it's time to come to a conclusion. Thank you.
Oh, that's just too bad. But the key conclusion is that numerous young factors, shown here in the red column, were upregulated in old animals or people through the neutral blood exchange or therapeutic plasma exchange. So one does not need to add them back. They are already robustly present once you calibrate the old factors. And then my conclusion is here is that there are many different attributes of aging, and yet there is a skeleton key which normalizes all of them as needed for healthy tissue. As I mentioned in the beginning, none of that could happen if but old mice become younger with respect to their cognitive capacity. So these are my overall conclusions, and thanks to the laboratory, as I mentioned, we are now undertaking phase 2 clinical trials with Dobry Kiprov being clinical director, and we are doing scientific part of the trial. So the conclusions are that young animals with their young blood and cells are rapidly aged by a transient exposure to old blood. Will young blood or factors be effective in old? Question mark. And then large dilution of systemic milieu stabilizes molecular and genetic determinants to younger states, and that makes most or all organs and tissues younger. Here are my disclosures. I'm trying to start the company. There are clinical trials underway, and we have been very, very fortunate with funding from NIH, state funding, and philanthropic funding. And thank you to the Canbo Laboratory. Sorry, I ran a little bit over.
Thank you so much for your lecture, Irina. Please answer some of the questions in the chat, but we don't really have time to answer questions on screen. So I think it's time to move to the next speaker. Thanks again for your lecture. The next speaker will be Her Fahy. You're ready, Her?
Are you able to see the talk? I'm not. I hit share screen, but I don't see anything happening.
At the bottom, there should be share screen.
I did that. I'm not sure that anything happened.
Currently, nothing is happening.
Why not? I'll try again.
There's a button at the bottom of that box, dialog box, you need to press to make sure that the sharing actually happens.
Oh, I see the share screen button, but clicking that doesn't have any effect. Oh, I see. Maybe there. Does that work?
Yes.
Super.
Okay. All right. Sorry about that. Okay. So let's start. Okay, so I know we're running a little late. I actually took Virginie's request to keep my talk down to 10 minutes to heart, so I moved several slides out of the presentation, so hopefully we can make up a little time. So at Intervene Immune, we're focused on restoring immune system function and reversing epigenetic aging in humans. So our goal is kind of symbolized by this photograph. As we go from left to right in the photograph, that's what we're trying to do. And I borrowed that photograph from a cover of Life magazine. So we focused on clinical trials because humans are the best model of human aging intervention. And it used to be that this is a real problem, partly because in order to find out if your aging intervention worked or not, you had to wait for everybody to die and then see if lifespan was extended. But we don't have to wait for that anymore because people like Steve Horvath in particular and other people who have been inspired by him have come up with global measurements of aging that are pretty good biological surrogates of aging in general. So we can use those kinds of endpoints to measure aging in humans and find out if an intervention is working. And as you just heard from Irina and also from Brian, there are now interventions that are safe enough and promising enough to try in humans. And so that's what we're doing. And you can study rats your whole life, but I think it's time to move on to humans because there's really no time to lose. We're all aging at a high rate, and many other people are as well, of course, and they need our help. So our focus has been on the thymus, which, as you all know, is the master gland of the immune system. It generates T cells that are important for fighting off cancer cells and invading bacteria and viruses. And unfortunately, the thymus involutes starting around the time of puberty. And this has a delayed effect such that somewhere between the ages of 60 and 80, we lose about 98% of our ability to recognize foreign antigens. And as a result of that, we lose immune system effectiveness over time. And just as we fall off the cliff on the left, we climb up this other curve in the middle, which signifies a risk of death. So risk of death tends to double within 2 years if your immune system is failing. Now, this is the significance of this has escaped people for many decades because the effect is so distant in time from the cause. The thymic involution happens around 12 or so, at least it begins there, but your immune system doesn't collapse until you're 65. But now we're all quite familiar with the importance of the immune system because of COVID-19, in which you see that if you're over 80, your risk of dying of this disease is more than 100 times greater than your risk if you're 10. And the reason for that is obviously because your immune system is aged. The good news about all of this is that involution of the thymus is developmentally programmed. In other words, it's caused by specific biochemical factors, and therefore those factors can be altered and used to reverse thymic involution. Although it's possible that maybe something like Irina's been working on could have some effect on the immune system in general, the thymus involution problem arises because of something that happens in youth. And so targeting aging is not going to really address the thymic involution problem. It has to be addressed specifically, and that's why we think what we're trying to do is relevant to combating aging. So we saw an opportunity to regenerate the thymus based on both animal and diseased human studies. What you see on the left is some of the results presented by Kelly et al. In 1986 in PNAS, showing that implanting cells that produce growth hormone led to rejuvenation both of the structure of the thymus and also the function of the thymus. And in the middle, you see a human study that was published in 2008 by Laura Napolitano, who was involved in treatment of HIV. And they gave growth hormone to their HIV patients and showed even within 6 months that they could demonstrate thymic regeneration based on CT imaging, and that this helped to replete the depleted naive CD4 and CD8 cell populations. In terms of normal people, and that's what our goal is, we want to apply this kind of technology to normal aging, not to some kind of disease model. That's my thymus in the upper right, before and after just a month on growth hormone therapy, and you can see an increase in dark mass indicated by the arrow, which is presumably an increase in thymic functional mass, which is demonstrably more than 2 standard deviations above the mean, and therefore a true sign of thymic regeneration in myself. So based on all of that, we started this trial called the TRIM trial, which stands for Thymus Regeneration, Immunorestoration, and Insulin Mitigation. We treated men who were 65 years of age or less with growth hormone between 2015 and 2017. We published results in 2019. This was a pilot feasibility, safety, and dose ranging study. We wanted to see if we could regrow the thymus just before the age at which the immune system collapses, so we could rescue people from falling off that immunological cliff. We used MRI quantitation of thymic fat content thanks to protocols developed for us at Stanford University, which is where the trial was based. We were able to collaborate with Steve Horvath at UCLA, with the University of British Columbia to ensure that we could measure epigenetic aging in our volunteers. And the treatment lasted for 1 year, was done under an IND from the FDA, was IRB approved. And by the way, we only had 9 guys in the trial, which is a weakness of the trial, but you'll see that we got robust results anyway. The core of the treatment is use of recombinant human growth hormone as a thymotrophic agent. It's given by injection 4 times a week using a special injection device that has growth hormone cartridges that go into it. The manufacturer does not make placebo cartridges, so it was actually not possible to have a placebo control for growth hormone in the study. The other component of the treatment was 2 anti-hyperinsulinemic agents, DHA and metformin. We use those because growth hormone administration elevates insulin levels. Insulin is a pro-aging factor and you don't want to raise it. So reference points there in the Napolitano 2008 HIV study shows that in her dose range, you expect to see a large increase in insulin. But I published in 2010 the fact that DHEA administration can reverse the hyperinsulinemic effect of growth hormone. So that was one of the components of our cocktail. It's advantageous also because DHEA declines with aging, so replacing it has a certain rationale. And the depletion of DHEA with aging is associated with increased all-cause mortality and various age-related problems. The second component is metformin. Obviously, it increases insulin sensitivity, so it will tend to combat this hyperinsulinemic effect. The Spindler lab showed that it's a very good calorie restriction mimetic, and you'll hear a lot more about metformin and its virtues from Nir Barzilai's presentation. Suffice it to say, we thought it was a strategic agent to use to block the side effect of growth hormone. So what did we see? First of all, we did see radiological evidence for thymic regeneration. On the left, you see the fairly white-looking thymus, which is mostly fat, becoming much grayer. In other words, the fat content is being replaced with functional mass. And as you can see in the middle, the probability associated with that change was highly statistically significant. We also looked at sternal bone marrow fat content as a control, and we found that that also improved by about 30%, again with high statistical significance, which is relevant because bone marrow function is important for aging and for thymus regeneration and for immune repletion. In terms of immunological effects, we saw recent thymic emigrants begin to come back. We also saw a repletion of naive CD4 and CD8 T cells. We also saw a reduction in exhausted CD8 cells, as indicated by this marker PD1, which has an additional point of significance about it, which is that PD1 is a checkpoint inhibitor, and lowering PD1 ought to improve the body's ability to recognize and combat cancer. So other results included, for example, a higher lymphocyte to monocyte ratio, which independent studies have linked to improved outcomes for at least 18 kinds of cancer and various age-related conditions, which are listed. And along the bottom, you see that our treatment lowered inflammation, at least as measured by CRP levels. It reduced prostate cancer risk based on PSA scoring. It increased FGF21, which is not only a thymotrophic factor, but also extends longevity in mice. It also improved kidney function, evidently, and we accomplished all of this without too much of a problem with insulin. Insulin did go up a bit, but we stayed within well within the normal range. Interestingly enough, we weren't expecting this, but some of our guys had their hair start to grow in dark again. So next time we do the study, we're going to have to pay more attention to that. The thing that caught a lot of people's attention was that we've also found that epigenetic aging seemed to be reversed by our treatment. Across the top, you see 4 different aging clock results: the original Horvath DNA methylation clock, Levine's pheno age clock, the Hannum clock, and then this more recent Grimm age clock. And as you can see in all cases, the change was highly significant. And in the case of the Hannum clock, and particularly the Grimm age clock, the change was permanent. 6 months after we discontinued treatment, we did not see any reduction in this improved epigenetic age based on Grimm age or Hannum age. And that's significant because Grimm age is a predictor of human lifespan. So we saw about a 2.2-year reduction in Grimm age, which would predict a 2.2-year increase in life expectancy. Of course, we don't have any substantiation for that, but it's an interesting observation. The average of all of these clocks is shown in the lower left and the turquoise points. And if you look at the slopes of the lines, it seems that epigenetic aging reversal accelerated greatly between 9 and 12 months of treatment. So up to 9 months, we were regressing epigenetic age about 1.5 times faster than chronological time was going by. But in the last 3 months, we were regressing epigenetic age at the rate of about 6.5 years of epigenetic aging reversal for every 1 year of treatment. This reminds me of this concept of Aubrey Gray's of longevity escape velocity, which with normal aging, you get 1 year older after a year, but if you're going to achieve longevity escape velocity, you either don't get older or you get younger, and we're firmly in the younger camp. You know, the guys ended up the trial on average 1.5 years younger epigenetically than when they started, or 2.5 years younger than if they had done nothing. So we're going to follow up on this trial. As I said, we only had 9 guys in the trial. TRIM X, which is the extension of the TRIM trial, we'll have, we hope, somewhere between 70 and 100 volunteers. We'll include women, minorities, and an expanded age range, anywhere from 40 to 80 years of age. We're going to have some groups that use growth hormone and some groups that don't, so we can parse out what the effect of growth hormone is with respect to all of these results. We also hope to have an untreated control group that will control for the effects of metformin and DHEA. We hope to include some T-cell functional assays. We're presently seeking grant support to underwrite some of the costs of the trial. One of the things that we hope to get out of this trial, speaking about biomarkers, is epigenetic aging reversal as an FDA approvable endpoint, which would benefit not only our own progress, but also everybody else who's able to reverse epigenetic aging by any means that they may apply. So in summary, it looks to us, at least based on our preliminary results, that epigenetic aging reversal and reversal of immunosenescent trends appears to be possible in humans now. And we hope to begin reproducing and extending these trials very soon and investigating mechanisms underlying the effects that we saw. And that's our contact information at the bottom if anyone has any questions. So thanks for your attention.
Okay, thank you very much for your interesting lecture. I think we can have maybe a question. Let's see what all we have here. So someone asked, since the thymus involutes quite early in life, but the T-cell reservoir stays strong until the age of about 60, are there compensatory mechanisms taking place in the meantime?
There are, yes, there are compensatory mechanisms. There's a lot of T-cell division that takes place in the periphery to maintain T-cell levels. However, it's only partially compensatory because that happens at the expense of genetic diversity of those T cells. Eventually, you run out of epigenetic, I'm using the wrong word, you run out of T cell receptor repertoire. In other words, the versatility of the immune system, the ability to recognize antigens goes down, and that's what that immune cliff was. But yes, that you can stave that off for quite a long time because you actually have the ability to recognize quite a large number of antigens when you're young. You can lose a lot of that before it starts affecting our mortality and morbidity. But if you look at the change in susceptibility to COVID, and look at the change in susceptibility to death in general, they match up pretty well. So we think that immune dysfunction creeps up on you from the beginning. It just gets worse and worse. But the real major impact is seen later in life.
Okay, and Jos asked, what do you think about FOXN1 therapies for the thymus?
Yeah, we think that we're activating FOXN1 with our treatment. There are various signs of that. If you look at the way FOXN1 affects the thymus and the way our treatment affects the thymus, they seem to be very similar. And FOXN1 might be predicted to do things like induce repigmentation of hair, which is one of the symptoms that we saw. So yes, FOXN1 gene therapy would be nice eventually, but as you know, that's a very long road and we all need something right now. We'd like to get there eventually, but for right now, we think we're getting there indirectly using these more normal physiological approaches.
Okay, thank you very much. And there are some more questions in the chat, so you can answer them there. Thank you for your interesting lecture. And we move on to the next speaker, which is Jos Mitteldorf.
How are we doing? Can you see me?
Yes, we can see you, but we can't see your slides yet.
I don't have slides.
Oh, okay. That's
you get to watch me talk. Just I'm going to ask for a lot of listening because ask you to think about some new ideas. I'm really grateful for the opportunity to be here today. This is such a prestigious and impressive group. I'm honored to be a part of it. For your interest and by way of apology, I'm fasting. I'm on day 4 of a fast. Sometimes this slows me down. I stumble over words. It's part of my own anti-aging therapy. So to begin, I want to talk about my favorite topic, which is the Data Beta project. It's about to get off the ground after long delays. We're hoping to start recruiting subjects in November. This is a community-based study designed to screen for anti-aging interventions already in use that have large effects, at least 20 years of life extension. So 20 years, you might say, if such things exist, we'd certainly know about them already. I'd say not necessarily. This field is moving very fast. The number of strategies that have been introduced in the last few years is large. They were unknown before. And also, this is something that sets our study apart. Data Beta is designed to look for combinations of known supplements and diets that synergize together. The premise is that most supplements and diets are based on a small number of pathways, so their action is probably redundant. For example, if we're taking metformin and also fasting, we don't get extra life extension from one compared to the other. But maybe hidden among these combinations, we'll find some unexpected synergies, the opposite of redundancy. So the combinations of strategies have extraordinary effect, perhaps far larger than the benefit you'd expect just by adding the separate benefit. We're looking for this, and no one else is. It's sort of standard in the field that we study one intervention at a time, and there's this hidden assumption that, well, they'll add together as if the effects were linear. And of course, that's not true. We all know that that's not true. So we'll be recruiting 5,000 people like you, biohackers, early adopters, people who are knowledgeable about the field and who are motivated to follow some combination of new and proven strategies for life extension. Every participant will fill out an extensive questionnaire so we know exactly what they're doing. We're not asking them to do anything different, just what they're doing already. And then we'll supply methylation testing at the beginning and the end of a 2-year trial period. We'll look for people who are aging very slowly, or maybe they're even younger at the end of 2 years than they are at the beginning. So our endpoint is the difference in age as measured by epigenetics at the beginning and the end. in analyzing the data, the tail of the curve is going to be most informative. This is the people who appear to be not aging at all or even aging backwards. What do these people have in common that we can learn from this? And if we find commonalities between the people who are aging slowest, then we'll say we've got something really interesting to report. This is a non-profit venture. There'll be no proprietary IP. I'll do the initial data analysis, but then all the data from the study will be uploaded to a website where it's publicly available. And I fully expect that other researchers will mine the data and find patterns that I've missed. So it's, in that sense, a large scale collaboration. Another sense in which is a collaboration, this is crowd-funded. Again, people like you, I've seen a lot of interest already, or maybe a major funder will step forward and boost the project along. In fact, progress thus far has been made possible by a generous startup grant from Didier, and I'm really grateful for that. And there's also been cooperation with my partners in this, my project manager, Walt Crompton, partners at McGill University, Moshe Sif, and my daughter, Sarah, who's injected some energy to push us over the starting line. So now for the main feature, which is about theory and biomarkers, which is what I signed up to talk about. Another name for biomarkers is surrogates. We need surrogate markers of age to evaluate candidate anti-aging interventions, because if we don't, then we either have to use short-lived animals or else we have to wait a very long time for human lifespans to play out. So there's a strong motivation to find surrogates. But then choosing the right biomarker becomes crucial. We want something that's highly correlated with age, but that's not enough. We really need to understand the fundamentals of aging before we can have confidence in any surrogate. So a trivial example, the lines in a person's face are pretty well correlated with age. But if we use this for a surrogate, then no one would say that getting a facelift was an anti-aging intervention. We're pretty confident that facelifts don't make people live longer. This is a trivial example, I know, but it helps us to think, why isn't wrinkled skin a suitable surrogate to evaluate aging interventions? Well, one answer might be because no one ever dies of wrinkled skin. People die of heart disease, cancer, and Alzheimer's, dementia. So we might be motivated to look for surrogates that are connected to these. Maybe arterial stiffness and HDL are markers for heart risk. Maybe cognitive decline is something we can measure as a marker for AD. Colon polyps, pap smears, PSA tests are markers for various kinds of cancers. But this gets very complicated, and we notice that all these problems are downstream results of aging, not upstream causes. So we're motivated to look upstream for causal factors that lead to all three of these things. For example, inflammation is measured by CRP and IL-6. Inflammation contributes to all diseases of aging. Decline of the immune function is also an upstream cause, and perhaps the loss of brain cells, muscle cells to apoptosis, this may be another upstream cause. Can we go further upstream? So this is why a theoretical understanding of aging becomes relevant. And I know we don't agree on what are the upstream causes, but it's something we ought to be talking about. I'm gonna go back to 8 years ago, 2012. I was one of several people scattered around the globe who started to speculate that changing gene expression was a primary upstream cause of aging. A very young Adiv Johnson and Joao Pedro, Tom Rando at Stanford, we all published separate papers proposing an epigenetic model for aging. It was well established and uncontroversial that growth and development were governed by gene expression, also known as epigenetics. And gene expression is under the body's control. The idea that the body is programmed to grow and for organs to specialize when we're young, well, that's natural enough. But the idea that this program might continue into a phase of self-destruction, as Vlagislani has theorized, this raised doubts in some people. At the same time, unbeknownst to those of us who were writing about gene expression and aging, Steve Horvath was putting the idea to the test. Methylation is one of the ways that the body controls gene expression. And as it turns out, methylation is relatively easy to measure in localized detail. A map, in fact, of what sites on each chromosome are methylated or unmethylated. Well, Steve got hold of several methylation databases with people of all ages, from in utero to over 100. He searched for methylation sites that are most tightly correlated with chronological age. There are 18 million methylation sites that are in the human genome, and of these, just a few 100,000 change consistently with age. He compiled a list of the best 353 sites that become either more or less methylated with age. And from these, he constructed what he called a methylation clock. This is a function that reliably and tightly correlates with age with an R of 96%. So what about that other 4%? Here's where I want to ask for your focused attention. Think about this for a moment with me. Horvath showed that the residual 4% was actually a predictor of future mortality and morbidity. The Horvath methylation clock was derived from computer algorithms that know nothing about biological age, know nothing about these markers. They only know the mathematics used only chronological age. And chronological age is, of course, a very strong predictor of mortality. But so we'd expect that the methylation clock would also be a good predictor of mortality. But the surprise here is that methylation age is actually a better predictor of mortality and morbidity than chronological age itself. So this 96% contains information about chronological age. The residual 4% is not just scatter. It actually contains information about the difference between biological and chronological age. That extra information about mortality and morbidity was not put into the algorithm by hand. It emerged from the mathematics. So the natural inference from this is that methylation is an upstream cause of aging. Methylation is an upstream cause of aging. The Horvath methylation clock is just what we were looking for, an upstream biomarker that can be used as a surrogate. And in other words, this confirmed the conjecture that João Pedro and Rando and myself were all making. The fact that the Horvath clock predicts mortality better than age itself means that methylation is a driver of aging. So this 2013 clock was already very valuable, but can we do even better? The methylation sites that comprise the Horvath clock must contain a lot of the drivers of aging, but I'm speculating that mixed in among those are some responses to aging that are not drivers. The theory is that as we get older, the body turns on self-destruction pathways like inflammation, like excess apoptosis, and it turns down protective pathways and repair like the immune functions, shutting down, stem cells become less viable. All this is driven by upstream methylation changes or drivers of aging. But at the same time, the body is responding, fighting with itself, trying to repair the damage that it's creating itself. And this comes from methylation changes too. And I call these downstream changes. They're responses to damage. So here's the crucial point in my take-home message. All these methylation changes, upstream and downstream, are useful if what you want to do is just measure somebody's biological age or their future risk. But if you want to evaluate an intervention to see if the body is getting younger, we want this surrogate to contain only the upstream drivers and not the downstream responses. Why is that? It's not just that the downstream changes are useless biomarkers like wrinkles or gray hair, they actually make our clock less useful for what we want it for. Let's say our body is making the body, let's say our intervention is making the body younger according to the drivers. That's what we want. Is it turning aging off at the source, shutting off the spigot of damage? But suppose our intervention makes the body, quote, younger according to the responses to aging. It's the opposite. We've actually shut down some mechanisms that are responding to aging and trying to rescue the body. With this context, I want to follow Horvath's next two ventures into to aging clocks. In 2018, with Morgan Levine as a student at the time, they designed together a procedure pretty close to what I think is ideal. First, a composite was constructed out of 10 markers of aging, including chronological age as one element, but also including inflammation, blood glucose, albumin, creatine, etc. This produced an aging score for each subject in the database, which Levine calls phenoage, and the methylation sites were chosen for inclusion in the clock according to how well they correlate with phenoage, the aging score. This clock was explicitly enriched in drivers. So phenoage predicts mortality and morbidity better than chronological age. And remarkably, the phenoage methylation clock predicts mortality and morbidity even better than phenoage itself. So phenoage is the raw markers. The phenoage clock is the methylation associated with these markers. And the phenoage methylation clock does better than phenoage as a predictor of mortality and morbidity. I think the phenoage clock is the best one that we have now. I wanted to talk about the Grim age clock, which I think is actually a step backwards. It illustrates a procedure that enriches the clock in responses to aging rather than drivers. For example, the Grim age clock depends on the smoking status of the person, and there's a methylation image of smoking, but you'd expect that smoking doesn't kill us by changing our methylation. It's killing us by destroying lungs and arteries. So the methylation changes associated with smoking are probably responses to aging, not drivers. And this is what we don't want in our aging clock used to evaluate anti-aging procedures. To close, I want to talk about what we can do in the future to do even better than the phenoage clock. Well, the phenoage procedures are, I think, just right. Start with a set of markers that we know are causes of aging. I'd recommend looking at inflammatory cytokines, NF-kappa B, looking for apoptosis, maybe P53, looking for mTOR, perhaps oncogenes, perhaps genes that are expressed only in stem cells, and look for methylation changes that are highly associated with all of these, rather than with responses to aging. And second, to improve on the Horvath clock, all the Horvath clocks are linear. They're designed to work from womb to tomb. And we know that the genes that are turned on and off during childhood are very different from those that are turned on and off later in life. We should have separate clocks for development and aging. Or if you like, we could have a single nonlinear algorithm that just uses different methylation sites at different ages. Now, this is a substantial opportunity for improvement. And I know people at Elysium have done that with their index clock. And third, there are separate positive coefficients and negatives coefficients in the Horvath clocks. We should optimize a methylation clock that just uses the positive coefficients and just the negative coefficients, and then combine those two later on. And this will prevent the cancellation of large positive terms with large negative terms, which I believe it throws a monkey wrench into the optimization process, which has been used so far. So in conclusion, the present generation of methylation clocks are already really good. They're getting better every year. And I'm confident we can make a methylation clock that's even better. And it will be everything that we need to evaluate candidate anti-aging interventions in the future. And that's what we all need. That's what we're here for. Thank you.
Okay, thank you so much, Jos. Let's see if there is some question. Wait, yeah, I think that there are some people who are asking, of course, if methylation drives aging, then what drives methylation? Is it the chicken or the egg problem we are running into here?
Since methylation happens in every cell in the body, there must be some, not there must be, I'm guessing that there's some central clock that controls aging, or perhaps the clock is methylation itself. That maybe that's, I'd say we don't know the answer to that, but people like the convoys and like the groups at Stanford looking at blood factors, I think maybe we'll have an answer to this question over coming years. If there is an upstream from the methylation changes, it's in the factors in the blood that are controlling methylation and other aspects of epigenetics.
Okay, thank you so much. Okay, we can go on to the next lecture. That's Ilya.
Once again, thank you very much for this opportunity. My topic is developing diagnostic evaluation criteria. This is absolutely necessary for any investment in longevity therapies. Okay, we know that if we really want to postpone age-related diseases, the main premise of anti-aging and of geroscience, of longevity, of the longevity field, that if we intervene into aging process, we can postpone the multiple aging-related disease or multimorbidity, and in this way extend the healthy lifespan. But what does it actually mean? What do we actually measure as a risk from aging? How do we calculate this? It is a question. And of course, we also know that there'll be massive savings from such an approach, from intervention to aging to prevent multiple disease. But once again, it's an open question. How do we calculate first of all, the effect on the actual health span, and then from this, the effects on the economy? It's very unclear. And we also know that there are multiple proofs of principle that it is indeed possible to intervene into aging, to postpone age-related disease, to extend healthy lifespan from animal models as well as from some emerging human trials. There is a multitude of approaches, of classifications of aging processes that are targeted. Once again, what do we mean by by biomarkers of aging. What do we select? Because obviously everything changes with age. Do we use these 7 processes, or do we measure some other 7 processes of 77 processes? Do we use this proprietary algorithm or another? It's very open, so everybody here is for himself for now. There's even a question. Of course, there are emerging biomarkers, a multitude of them. Once again, everything changes with age, so everything can be a biomarker. There is no consensus and no clear vision. What is it that we're actually looking for? No consensus, no agreement even about the most basic definitions. How do we measure aging? How do we see aging? Is it a disease? Is it a syndrome, polysyndrome, risk factor, underlying cause, frailty predictor? Do we measure biological age? Do we measure multimorbidity? Do we measure index? What do we want to measure? So far, there's no agreement, but there are some operational approaches, some instrumental approaches that allow us to function in real life. Probably the most practical is the approach by Nir Barzilai, who is here, the targeting aging with metformin. So if we don't know how to measure aging, at least we can diagnose multiple diseases, combine them, and this way measure the effects. This is probably the most practical, feasible approach right now that was accepted by the FDA. And there are already some ramifications, some additional applications of this approach. For example, here in Israel, we introduced this approach into the call for research proposals on aging called the Britain-Israel Research and Academic Exchange Partnership on Aging, BAREX Aging, that explicitly seeks approaches toward early diagnosis and prediction of old age multimorbidity and aging as determinant of multimorbidity. Over 200 labs submitted proposals. So it's very far from common practice, very far from good practice, but at least more and more people are exposed to this approach of measuring multiple disease, measuring the aging process. It is also very important to introduce this topic into policy. That's my focus. I'm, half scientist or half part scientist, half-time activist. For example, in the position paper we wrote in the International Society on Aging and Disease for the critical need to promote research of aging. One of the central topics is developing criteria for efficacy and safety of geroprotective therapies. It was translated to 12 languages, submitted to several governments, quoted 150 times in academic literature. So if we may not agree yet on concrete biomarkers that we want to use, at least we can agree on policies and introduce this as a policy that governments, that authorities should pursue. Also, in the international frameworks, we need to push for this kind of strategic vision to develop aging biomarkers or multimorbidity biomarkers. Some of the examples where our advocacy was actually somewhat influential. First of all, the famous case with ICD-11, where thanks to the work of Daria Khalturi and her colleagues, they included aging as a qualifier, as an extension code, aging related, into the ICD, into the International Classification of Disease. But once again, aging was in the ICD even before as senility, old age, but there were no agreed biomarkers, so it was considered a junk code. So before we agree on some acceptable biomarkers, there's no way we can apply this criteria. And also in the campaign we did in 2017 to introduce aging into the work program of the WHO, we were successful. Aging was included with some hints at measurements. For example, they say that they should measure healthy life expectancy as the main measure of the effectiveness of healthcare system. They say that we should reduce the number of care-dependent elderly by 15 million. But what does it mean? Nobody knows. Where are those 15 millions in Western countries, Eastern countries? How do we measure age care dependency? Generally, we need to know that WHO is not so great on evaluation. For example, if you look at the global burden of disease, the way they establish risk factors for death, it turns out that we all have many hundreds of % chances of dying. when in reality we still have only 100%. So we really need some good methodologies to evaluate risks, to evaluate biomarkers. At least we need to include this search. Here in Israel, we also push to include it into policy, once again, not as a concrete biomarker or set of biomarkers, but as a strategic part. In 2018, we were able, together with my comrades, Alex Friedman from Disabled Not Half a Person, were able to introduce this topic as part of the Israel National Master Plan on Aging. First of all, we were able to introduce this very topic of education, research and development for health and longevity and preventing age-related diseases, the National Master Plan on Aging. And within it, one of the topics was to establish infrastructure for preventive measures for aging-related diseases. But this basically means developing and implementing biomarkers, appropriate biomarkers on aging. It still sits very nicely on paper. Didn't advance far, but especially now with the current situation, also political situation in Israel. But we're hopeful that it will develop and become a project potential working group. Our research, our research together with my colleague, Dr. David Bloch, we also try to work on this topic, by our own means. We use information theoretical measures such as normalized mutual information, and it allows some new approaches to measure multimorbidity and aging. For example, it allows the establishment of non-linear relation between parameters, and also the establishment of cumulative or synergistic relation between parameters. Josh also noted many processes in aging are not reducible to linear relations, also not to single cause effect, but also synergistic. This approach allows to measure such relations. We published some papers in good journals like Progress in Neurobiology, even introduced this topic as a commitment in the European Partnership on Active and Healthy Aging. But once again, it's only our project that not many people heard about. There are thousands of other projects running in parallel about biomarkers that probably never heard about each other. We need some kind of dialogue. We need to come to some common denominator, probably not about specific biomarkers, but at least about some things that we're looking for in a biomarker. Is it clarity of definition? Is it diminishment of confounding factors? Is it informative value? Is it practical utility? Is it cost effectiveness, affordability? Several criteria that can be explored and to drive our search for acceptable biomarkers. And of course, it is important that the established set of biomarkers is used in regulation to actually approve therapies. Otherwise, it will once again remain on paper, but also that it should not stifle the emergence of new biomarkers, because obviously there will be new and more effective biomarkers probably every week or every month, and we should develop our evaluation criteria in such a way that will not stop the new developments, but only encourage them, encourage competition. Once again, competition is great, multiple approaches are great, but we also need some kind of a common language to discuss. Of course, in physics, you know what different concepts mean, what force means, and how we measure it, and how we do tests with it. In aging, we are very far from it, and hopefully in a few years or decades, we'll get to that language for our conception. What we can do now is really to encourage consultation on the subject, to encourage opinions, exchange of opinions, some sort of consensus forming. We could really strive to sort of, guidelines. For example, together with Alexei Moskalev, we are now editing a special issue in Frontiers in Genetics, exactly on this issue, clinical evaluation criteria for aging. So we encourage everybody to submit their opinion articles or reviews or some concrete experimental studies. We really need to increase public discussion of this issue. Otherwise, we really have no meaning to anti-aging treatments. Not treat something that we cannot diagnose. Very major bottleneck in longevity research. Ideally, there should be some authoritative working group with some authoritative governmental or super-governmental agency that will sit down and discuss and bring different opinions, do meta-analysis, and come up with some guidelines like they do with the ICH. But it costs money, and people have their jobs, and not everybody would be available for such working group. These are some of the agencies that at least I know when this topic was broached. At least there were some attempts to establish some sort of working group. Nothing succeeded. All these attempts failed. But hopefully, perhaps thanks to this conference and similar approaches, there will be a more ongoing, sustained effort to come to the common ground to develop some common guidelines for biomarkers development and application. With that, I thank you for this.
Okay, thank you very much for your interesting talk. I don't see any questions currently for you. So
there's one from Jose. Jose is nice to do.
Oh, okay.
I know there's just thanks. No, no question. Yeah.
Yes, and actually, I would like to ask everyone to do exactly as Jose. And in the future, when you have a question for a speaker, you just put the name of the speaker first. That makes it easier for us to find the questions in between all the dialogue that is going on in the chat as well. Okay, so the next speaker is Nir, and I give him the word.
Thank you very much. Thank you for the organizing. Thank you for really important and challenging talks. So my title was actually longevity genes. And in the last 48 hours, I was asked to talk about 4 other topics as well within this time frame. So all I can do, and some of you before have assumed what I'm going to talk about. So what I'm going to do, I'm really going to make some provocations. That's all. I'm going to talk about 3-4 things and just make the provocations that I think are more important. And I do want to start with longevity genes, and this is our poster family. Those are all siblings that live to be between 102 and 110 years old, to show the effect of genetics on longevity. We have more than 750 centenarians in our study now. We have offspring and controls, so we can do a lot with our studies. But I think the main thing that we answered first, the question was, are they getting sick when everybody gets sick? And now they live sick for 40 additional years, or is their lifespan and health span goes together? And this is a paper together with Tom Pearl's group that shows that centenarians actually live 20, 30 years more healthier than other people. So it's about health span, and we know that there are those centenarians whose health span has been increased significant, or we could say their aging has been slowed significant. And in fact, the most important thing about them is not only that they live healthier, but they had contraction of morbidity at the end of their life. They lived, and basically died within few weeks or months. Even the CDC, we know a lot about the CDC recently. They looked at the medical cost of the last 2 years of life of somebody who dies over the age of 100 or 70, and it's third the cost. Okay, so we know that achieving health span and is associated with what we call a longevity dividend, and that it's worth looking. But our biological mission was to find those longevity genes and develop drugs that will affect them. And this is, I took many slides that I had and just made one slide to tell you that in all longevity around the world, the genotypes of ApoE2, okay, not ApoE4 of Alzheimer, but and not ApoE3, but ApoE2 is associated with exceptional longevity, and there's probably a biology behind it. And there's a specific genotypes of FOXO3A that came from the East, actually, that was validated many times over and seems to give some protection against aging. Those are public, but when you go into population, you can see also specific genotypes. CTP and ApoC3 are not common to everyone in the world, but their population in the world, that if they have this specific genotype, they live longer and healthier. In fact, CTP and ApoC3 are example of few genotypes that pharmaceutical went and developed antagonism against them and went through phase 3 to show that they are working. So our research have really been already translating. It's Merck for CTP and Ionis for ApoC3. One of the mistakes that people have is that they think that, well, there's one longevity gene for everybody who lives above age 100. And in fact, where the research is and where we are at is that it's not about a common genotype, it's about clusters of genotypes in our population that have a functional consequence, and look at them and see how they are related to aging and longevity. Just as an example, 2% of our centenarians have functional mutation in the IGF-1 receptor, okay? With this, we went back into the lab and we used IGF-1 receptor antibody and showed that aging can be, that health span can be extended and lifespan can be extended with this antibody. So the idea of having centenarians as rare population where aging is slowed is really picking up. And in fact, Regeneron and AFAR are going to announce soon launching a project of getting 10,000 centenarians, hopefully from all over the world, in order to get just more statistics, more power, and finding everything important that we can find about this population. The last thing I want to say about these genetics is that the question was always, is longevity in humans similar to longevity in animals? Because everybody complains that the models that we use, the mice models that we used in the lab were not predictive of any treatment in humans. And we did so we have exome sequencing of all our population. And basically what we've done with Zhengdong Zhang is look at at rare genotypes. We're talking about less than 1% of our centenarians. So we have basically, somebody who has 16, centenarians have 16 variants and control have 0 or 1, those kinds of data. But put them into pathways, okay? Put them into pathways and get some more information about the pathways and see what's coming up. And what's coming up is really incredible. What we're getting is exactly what we know from animal, okay? Like MIP kinase, like insulin IGF signaling pathway and mTOR signaling pathway, they're the top pathways that are coming out. So I'm going to leave human genetics, but longevity, exceptional longevity is a good way to look at practically about aging and what can be targeted. And yes, when we're doing those studies, we are getting the results of the animals because our whole concept was that aging is conserved. Okay, I'm moving out to make a comment about biomarkers. And remember that the most important thing of biomarkers is that they change with treatment. And the sooner the change, the better. We don't want to wait months or years for a biomarker to change. We want to do study that we will get result immediately and know what's going on. And this is limitation in many ways of the methylation. The methylation in our hands is very stable. It doesn't go that fast. And actually the studies, we saw the only good example of 9 people, but the studies are not really that great, or they cannot be the only thing we trust. And what we did is we collaborated with Somology on our study, and never mind the technology, but we're measuring 5,000 proteins in 1,000 people between the age 65 and 95. And we're basically asking what changes between 65 and 95. We had a Nature Medicine paper collaboration with other groups that looked at all ages, but you know what? Everything really happens between 65 and 95. We have 2 additional aging cell papers coming out soon about this proteome. But the bottom line is, this is a volcano plot. I'm not going to educate you too much, but those hot lava plot, the reds, if they're going high enough, this is the p-value. So we're talking about 10 to the minus 80. And if they're going far enough, like with BNP, that's how effective it is. And what we see is that we have lots of significant proteins. And as Josh said before, you know, those proteins, some of them may be cause aging, but some of them might be protective against aging, and we have to distinguish between them. And here we found something really interesting and very relevant for a biomarker that change with treatment. When we do a pathway analysis, and there's many ways to do pathway analysis, on one hand, the most significant pathway is the insulin growth factor pathway. It's really interesting. But what you see here in reds are all breakdown products. There's breakdown of the extracellular matrix, of collagen, of neutrophil degranulation, of platelets degranulation. And at first I said, what is this noise? I don't learn anything from a breakdown programs. But then I realized, you know, no matter how we're going to treat aging, we have to stop this breakdown. Okay, I think this breakdown is going to be extremely important proteomic markers, and we should show in therapy that we're stopping this breakdown no matter what we do. I think it's can be a major component of proteomics for aging. I would just tell you in our studies that those 1,000 people, half of them were people out of the community without family history of longevity, and half of them were people who are children of centenarians, and we showed that they are much healthier. And what you see here, you see that it's red here and much more white here because while our offspring had 585 proteins that were significantly changed, the offspring had only 235. In other words, they are still younger. You know, they'll get there when they'll get to their biological age. And in fact, what we're finding with them is that they have 29 proteins that the others don't have. And some of those proteins seems to be protective against aging also. So there's a discovery part here. I also want to say something else that somebody said before, or one of the comments. Female, you know, male have 600 proteins that are changing, and females have 250 proteins that are changing. The proteome of the women is much more stable. And we have to recognize that. We have to do age-specific in aging. There's no way around it, even when it comes to biomarker. And of course, we can build the clocks, and the clock that we've built showed that our biological age by proteomic is much better than chronological age and even frailty. So those are the kind of things that we want to know and want to follow. So this is the provocation about proteomic. They need to reflect treatment. We want to see fast effect of treatment. Proteomics are going to be important. Now, the challenge that we have, and that was mentioned before, is that aging is not known as an indication. And it's a problem. It's a problem because we need somebody to agree that aging can be prevented. That's the best we want to do, that aging can be prevented. and that there is an indication for prevention, and that pharmaceutical will jump up to the increasing number of biotechs and start collaborating and funding a lot of what's going on there. So can we do something about the regulation? And the answer is that we found a terrific tool to use, and this is metformin, because metformin extends a lifespan and health span in animals. And we recently showed, those are here, down here are all the hallmarks of aging. And we showed that no matter, that when we looked at the literature, metformin seems to affect all the hallmarks of aging. Now, I don't really believe that in the sense that there's a direct effect. But it's common to every drug that you intervene, with sirtuin, with resveratrol, right? With rapamycin, with metformin. If you really target aging, you're going, let's talk about the cell that now is becoming younger. Well, you're going to fix lots of things. Okay, so at the end, you get a lot of effect, and we can argue which is primary and which is secondary. And I don't know what metformin is doing first, okay? But at the end, we can see the whole hallmarks of aging being affected. And that's why one of the interesting thing about the hallmark of aging is there's the connectivity between them. We know that we target one and we approve the other. It's because we're doing something significant about aging. And of course, we know from both clinical and association studies that metformin prevent diabetes and prevent cardiovascular disease, that people with metformin have much less cancer. There's clinical study, one recent, that shows that people on metformin have less Alzheimer and less MCI or progression of MCI. And, you know, I think it's really interesting from the UK, this study that showed that people on metformin who have diabetes and are obese and are sick to begin with have less mortality than people who were treating by the same doctors, the same pharmacies, who don't have diabetes. Okay, so you can see that you can actually measure metformin's effect on many outcomes, or on cluster of outcomes that include mortality. We are talking about a drug that's really cheap, safe. As I said, we're using it more as a tool because we need to repurpose a drug in order to just basically show that the concept that we have is working. And the way that we're going to do it is we're going to take people between 65 and 80 years old. We don't care what diseases they have and what diseases they're going to get. That's not the point. We're saying no matter what you have, we're going to, if you're on metformin, it's going to get later. You're going to get it later. So there are 3,000 people, half of them are on placebo, and we have clinical outcomes that the FDA want, and we are supported for the biomarker part by the NIA grant. Unfortunately, TAME was on hold because of many reasons. The most important is the COVID. And I'll tell you, I feel lucky that we didn't start TAME last year, because it would have been a disaster now. But we're ready to move whenever COVID allows us. So I think I took a lot of time. Let me stop here, and I'll be happy to answer question now or later at the web. Thank you for listening.
Thank you so much for your interesting talk. I would actually like to start with a question of my own. So you published a paper in Nature Medicine last year showing that the plasma changes in the proteins happen in like these 3 waves. And I wonder if you have any idea of what could be the reason it happens in waves and not in a more continuous fashion.
Yeah, so that's a great question. Look, I don't see aging as linear, and we have to know that, when we're talking about walking speed or, hand grip and stuff, this is going to be stable for lots of decades, okay? Much more stable than what happens between 65 and 80 years old. So I don't think that aging is linear. I also don't think, in fact, I know those proteomics that I showed you are breakdown. They don't happen in young people. Most of them are not happening. The overlap is 1%. They don't happen in young people. So this aging is accelerating at age 65. We're starting to have this breakdown, and we're starting to have the other things, and we have to realize that that is the point where we have to intervene, and we know what we're looking at.
Okay. Well, I see that actually 2 people at least are asking about pain. So do you have an idea when it will start? Will it be immediately after COVID is controlled?
It's we say here it's hard to make predictions, in particular about the future, right? But we have 14 centers around the United States. And in my view, the New York, the Einstein and the Harvard Center are kind of ready to go. We're starting to open everything, so it is possible that we'll stagger, but we want to start. Connecticut also actually has a center. So I think we can starting where we can start, and we're adjusting our protocol from point of view of visits. We are all doing it. We can have really good control by electronics these days, and we can manage the visits much differently than we used to before.
Okay, thank you so much for your lecture. We are going to have a pause now. There are some more questions in the chat, so you can answer them there. Thanks again for the lecture, and let's take 10 minutes break. Get some coffee, and I see you back in 10 minutes.
And have yet to find an actual change. We're discovering new and ingenious ways to solve problems that were once thought impossible, like manned flight, instant global communication, powerful computation, spaceflight, and advanced medicine. We dream of a brighter future. And let's face it, we all like to be healthy. We generally don't like being sick. We enjoy doing the things that healthy minds and bodies allow us all to do. Though invariably at some stage we become less physically and mentally capable of doing some of the things that we like to do. Our bodies become less capable of dealing with ailments, and we suffer under the burden of disease, the spectre of 1,000 faces. We have a choice of mildly prolonging the good life by putting the odds in our favour through a balanced, healthy diet, regular exercise, and visits to the doctor. History shows that on average, life expectancy has been going up. And there are promising developments in biotechnology and medicine which indicate this trend will continue. However, the fundamental problem of aging remains. No matter how good we are to our bodies, as a byproduct of aging, we just wear out. Our bodies become less capable of dealing with disease, and our quality of life declines. What would we do if we thought that it was possible to cure aging within our lifetime? We know there are some animal species that can live a lot longer than humans, like the Galapagos tortoise. Also, some creatures have remarkable regenerative capabilities, like the axolotl, capable of growing lost limbs, tails, regenerating severe damage to other organs. Surprisingly, in fact, every species is capable of regeneration, from bacteria to humans. Unfortunately, unaided by appropriate treatment, humans are only capable of regenerating to a limited degree, like tissue regeneration. Throughout history, cultures from all over the world have the desire to achieve the means to defeat aging and conquer. Of youth appears almost everywhere in writings in Greece, by Herodotus to volumes of Chinese legends. and all the way back to 2100 BC, the ancient Mesopotamian Epic of Gilgamesh, one of the oldest stories ever told, though these are all, of course, myths and legends. In recent times, we have come to know a lot more about the world and have yet to find an actual fountain of youth. People could be forgiven for thinking that the ravages of old age are inevitable and accepted as just as part of being human, as part of the learning process of growing up, as an inescapable destiny. Seeking comfort from existential angst, some retreat to the standpoint that it's never going to be possible, therefore it's okay to be irrational about whether it's desirable. No, it's not okay to be irrational about this. Of course it's desirable, like generally reducing suffering is desirable. Many people's perspectives on aging are evolving. They see promise in advances in medicine and biotechnology, and in hope look forward to the possibility of having long, healthy lives. So what is aging? It turns out that people are pretty confused about aging, so let's look at how aging and disease are related. The word disease comes from Old French, meaning a lack of ease. And when you break up the word, it makes sense, dis-ease. The word age was imported from Latin into Old French and then into English. Its meaning is close to era, relating to the time somebody has existed. Throughout history, aging has been related to all sorts of ideas, mystical religious ideas. Aging is the source of meaning, a social identity, a personal quality or virtue, a foregone conclusion, or even a natural phenomenon that shouldn't be tampered with. This is strange. Aging is sometimes framed as natural and therefore sacred. There are some gaping holes in this reasoning process. The appeal to nature is an informal logical fallacy. The content of the argument fails to support its proposed conclusion. Not only is it difficult to define what natural actually means and where the boundary is between natural and not natural, but obviously that which is found in nature is not always good. Would one feel comfortable with endorsing infanticide, mass killings, rape, cannibalism, parasitism, just because we find examples of them in nature? Despite biogerontologists' best efforts, people seem to be under the trance of harboring two paradoxical positions when selectively appealing to nature when aging is brought up, but not when we're talking about natural diseases as byproducts of aging, which should be combated or eradicated. An honest observation of the suffering caused by the diseases of old age invokes unease. Is it ethically acceptable to not do anything about it? Well, the good news is people are doing something about it. Though given the scale of the problem, it seems there is a huge underinvestment in funding translational research to treat or prevent aging. So what is meant by aging is contextual. In the context of the health of the organism, or in a biological context or a medical context, the most appropriate definitions are precise and specific. How aging has been categorized and defined has influenced the way we think about it. On the one hand, we have our chronological age, which refers to the amount of time we've been alive. And on the other hand, we have our biological age, which refers to the condition of our bodies. Like cars, bodies eventually wear out, and without maintenance, they degrade, fall apart, and stop functioning. A side effect of driving our car around is that the tires bald, the brakes wear down, clutches burn out, radiators and piping leak and develop calcium deposits. If we keep our cars well serviced, giving it regular oil changes, mechanical cleanups and general checkups, our cars are expected to last a lot longer. And if done meticulously, then our cars can last indefinitely, well past the original intended lifespan. In a conceptually similar way, but in a very complex fashion, our bodies wear out, metabolize, malfunction, and eventually just stop working. Should we classify aging as a disease? If aging isn't considered a disease, it's easy to consider it as normal and not as a target for treatment. The harsh reality is that chronological aging and the diseases of biological aging are highly correlated. Biological aging will at some stage cause suffering, and a lot of it. Fortunately, there's been progress around categorizing aging as a disease. If you can classify aging as a disease, then you can treat it. The World Health Organization added a new extension code for aging-related diseases, defined as those caused by pathological processes which persistently lead to the loss of the organism's adaptation and progress in older ages. This will certainly help to overcome regulatory obstacles to achieving rejuvenation medicine. Why? Well, in the Lancet in Diabetes Endocrinology, there's an editorial published entitled Opening the Door to Treating Aging as a Disease. As ICD codes are prerequisite for the registration of all new drugs and therapies, the recognition of aging as a pathological process, together with the replacement of the ICD codes for senility with old age, represents undeniable progress towards overcoming regulatory obstacles that have thus far hampered the development of therapeutic interventions and preventative strategies targeting aging and age-related diseases. It is obvious for the most part that we love life so much we systematically try to avoid injury and death. People have been asking the question,
Now it is my turn, yes? And a great pleasure and great honor. It is a great pleasure and a great honor to start the next panel, the next session, which will be exactly on biomarkers of aging. And the first speaker is none other than Aubrey de Grey from Sense Research Foundation, who will speak on the importance of physical biomarkers for aging.
Ilya, thank you so much. And thanks to Sven and Didier and Virginie and all the other organizers. It's great to be back at EHA. I love speaking to this group, of course. Now, since I've only got 10 minutes or so, and since I tend, when I speak to this group, to not trot out my normal talk, I'm going to not use slides today. I'm just going to pontificate. As many of you will remember, typically when I speak to people who are familiar with Sense Foundation's work, I tend to try to give some kind of call for action. And the topic of today's conference actually gives me a good opportunity for that. The thing I want to focus on, and actually this is also somewhat serendipitous because the organizers actually chose the title of my talk for me, but it worked out rather well. The thing I want to focus on is what I view currently as a bit of a deficiency in the current work that's going on in relation to biomarkers of aging. And it's a deficiency which I think that many people here who may have computational expertise, even though you may not be professional gerontologists, could actually contribute to. So here goes. As all of us know, the idea of a biomarker of aging has been around a long time. And the goal of finding one has been one that everybody has recognized the importance of, if we could. In particular, the National Institute on Aging in the US, back in the 1970s and 80s, spent a lot of money, I mean a lot, in an attempt to identify biomarkers of aging that were actually reliable. And it was, I am not exaggerating by telling you that it was a complete bust. It was utterly unsuccessful. And basically people came back feeling that such a thing wasn't possible. Now, there were two reasons why their pessimism at that point might have been premature. One of them was that, to be honest, they hadn't really decided, at least not justifiably, on what the definition of a biomarker of aging even is. You know, some people would say, well, it's a predictor of how soon you're going to die. Some people would say, well, no, it's a predictor of how soon you're going to start getting sick from one or another of the classical progressive conditions of late life, whether mental or physical. Some people would say, well, you should measure it as a proportion of how long it is before you die, and so on. But there was really, not a lot of agreement there. However, the other big reason, of course, why there was perhaps premature pessimism was that back then we did not have big data in the way we have today in biology. We did not have the ability to develop the kinds of things that we're hearing so much about these days in relation to biomarkers of aging, specifically omic clocks of this or that type. The epigenetic clocks of Steve Horvath and Morgan Levine and others have, of course, got more publicity than any other, but they would be the first to agree that the best clocks that exist right now of that kind are multi-omic ones, or at least in due course there will be multi-omic ones that incorporate proteomic data, metabolomic data. So a lot of big data out there. So that's all very good, and of course it has restored people's optimism that we can, in due course anyway, become rather competent at predicting how long it's going to be before somebody gets sick. But there's a big difficulty here. If we ask ourselves, well, why do biomarkers of aging matter so much? It's not just observational. Back in another problem, of course, that exists in the gerontology community in the 70s and 80s and 90s, and perhaps even edging into this century, was that gerontologists were very pessimistic indeed about interventions. In fact, the culture was so negative that you couldn't even mention intervention in a grant application and have any hope of getting it funded. Whereas now, of course, the situation is pretty much 180 of that. It's the exact opposite, where you kind of have to talk about the translational potential of your work or else you don't get it funded. So that's a great thing, but what it should concentrate our minds on is the need for biomarkers of aging, not only to tell us how soon somebody is likely to get sick from a chronic condition of late life, but also the extent to which this or that intervention can alter one's propensity to get sick or the time frame before one's likely to get sick. And that is actually a lot more difficult, If we think about today's world in terms of the difficulties that I'm sure Nia spoke about and other people have probably spoken about already today, of getting drugs for aging through the regulatory obstacle course, we all know that one of the biggest difficulties of all is that aging is not typically considered to be a treatable condition. And of course, that is gradually changing as a result, not least of the sterling efforts of Nia and his colleagues in developing a kind of complex combinatorial definition of aging in all but name that has been incorporated as the clinical endpoint of the ATTAIN trial. But there's still a long way to go there. And the difficulty, of course, is that aging in humans is so slow. So if you want to validate a biomarker of aging in mice, no problem. You just see whether it correlates with how long it is until the mice actually get sick or die, because that's not going to be very long either way. But you can't really do that for humans. So, of course, what initially happened when people like Steve Horvath were developing their epigenetic clocks was that they simply developed a clock that was a really good correlative chronological age. That itself is, of course, completely useless because we already have birth certificates for that. But they knew that, of course. People like Steve knew that perfectly well. And so they moved on. And where we are now is clocks that purport to be clocks for biological age, demonstrators of biological age, which is, of course, the platonic ideal of a predictor of how soon you are going to get sick. All great so far, except when you start to bring interventions into that equation. Whenever you're trying to develop a statistical fit to data, when you have shall we say, a training set and a test set, you have a training set that you identify your weighted sum of this or that number of parameters, especially when you're talking about hundreds of parameters, then a huge danger, which everyone, of course, is very well aware of, is the danger of overfitting, of getting something that works really well for your training set, but actually does not work particularly well for new examples. And so people have got pretty good at avoiding overfitting. But there's a big catch there. The big catch is that overfitting is only avoidable if there is no particular reason to believe that your test set, your training set, is inherently different from the test set. And of course, there's a very, very important way for clinical purposes in which it is necessarily different. Namely, the training set consists of people who have not received therapies that don't yet exist, whereas the test set is going to be precisely people who are receiving a therapy that's just been created and that you want to determine the efficacy of. So what we need is biomarkers of aging that are robust to that, that with very high probability do vary with real biological age, whatever the intervention is that you throw at them. And that has to include interventions that don't yet exist. So that's a bit of a conundrum. That's inherently not possible, right? So how close can we get? Is there a way that we can get close? I believe there is, and I believe that it's a way that is not yet being adequately paid attention to. And moreover, since many of the data sets that are involved here are public, this is ultimately something that people with good access to computational resources and understanding of the problem can actually do. That means a lot of people that I'm speaking to right now. So what is this trick? What's the trick? What we can do is we can identify correlations between biomarkers of aging, such as an epigenetic clock, and other biomarkers of aging that have been established traditionally over the years. Now, when I talk about other biomarkers of aging, of course, I've already given the game away in a sense, because we know that those other biomarkers, those traditional ones, basically don't work very well. That's what I was mentioning earlier about the failed attempts back in the 1970s and 80s. But they still work fairly well. We all know that, it's awfully good if you don't have much insulin, and it's good if you have strong grip strength and gait speed. Things like this, there's some correlation. And those are things that you can measure at once. So what we actually need is a clock that is tested not only with a bunch of untreated people, but also is trained in clinical trials for interventions that work and for interventions that don't work. If we can go back and look at historical data on such things, and we can see whether, for example, grip strength was actually a good predictor in a particular clinical trial of whether the therapy would actually delay this or that aspect of age-related physiological decline, then we can also ask whether grip strength correlates with the epigenetic clock in the clinical trial. If we do that over a lot of trials, then we can develop a clock which correlates well with other individually less good biomarkers of aging. And I believe that that is something that fills this gap. It's something that has the opportunity to make such epigenetic or other omic clocks be much more robust and useful and relevant for determining the potential of a new intervention. Okay, I think I've probably run out of time already because I was rather long-winded about that. I hope I have got my concept across. I guess it was a bit sophisticated, but I hope the idea is clear. So I'll stop there and I'll answer any questions you have. Thank you.
Thank you very much, Aubrey. Which ask is
okay, Sven, you're breaking up, Sven, but I can see that Jose Cordero has a question in the chat. Perhaps that's the one I should be answering. Jose asks, do you expect to see many differences in epigenetic clocks across different mammalian species? Unfortunately, the answer is, oh yes. In fact, it's pretty much impossible to just transfer blindly across species. Now, of course, that's a good thing in the sense that kind of doesn't matter in the sense that you can always develop a clock in mice, and indeed Steve Horvath and others have already done that. But it does mean that we can't just use the clocks, develop the clocks in mice and just say, okay, let's use those clocks. You can't do that.
Okay. Other questions? Thank you very much, Aubrey. Then we go into the next speaker, Anton Kulaga from the Institute of Biochemistry of the Romanian Academy of Sciences, who will talk about federated learning on human data for biological age prediction. Please.
Okay, so do you see my slides? Okay, so thank you very much for the introduction. So today I will be talking on a simulation study that we are doing on applying federated learning technology on human data for age prediction. So the study is motivated actually by one simple question: why we see so many magnificent results of applying machine learning models to general problems like differentiating cats from dogs better than humans can do, while in the same time we see that in domains that matter, like biomedical domain, the results are extremely modest. And on the right picture, you can actually see an example of such type of mistakes. So if we look into the common workflow that is used when you work on machine learning problem, you can see that if you're working on a popular general problem, usually what you do, you download the data set, you train a model, you tune a model, and another CATS classifier is ready. While when you are working with some sensitive data, like biomedical problem data, before you can even download a data set, you actually have to acquire it. It means that you have to somehow purchase the data set from the hospital, and for this you probably need money. You have to get investors, make business plan, make business, spam everybody, or if you are an academic group, you have to make a consortium and win a grant. That is also quite hard. So the main problem is getting access is hard. And for this reason, most of us focus on solving tasks that are accessible and getting quite good results. But what about actually focusing on tasks that matter, like solving different diseases and addressing the main disease, aging? And in aging, what is special about is, as it affects all of us, it has one especially important particular property, is that you can take a clinical study on pretty much any disease, and you can take out all the healthy control from them. And you can use this data to train an aging classifier, because you can know the age of these people. That means that in theory, we can use pretty much any clinical data set to train an aging predictor. And so many thousands data sets can potentially be available for us. But the problem is that it's too many data sets, and we have a lot of regulations like GDPR, like HIPAA. And the trend is that regulations are getting tougher and tougher. And you can easily feel it by enormous amount of cookies that you have to accept when you surf web. And many people use technology like cookies blockers or acceptors in order to make web surfing great again. And for sensitive data, things are even harder and become even tougher. So we also need technologies to make data science and machine learning great again for biomedical data. And there are 5 core privacy preserving AI technologies. So by privacy preserving, I mean that you can train a machine learning model on the data that you cannot actually see, where you cannot see individual patients. So two last of them actually require some hardware and are pretty slow. So we looked into first 3 of them, and for synthetic data and federated learning, we trained some models in order to do comparisons. So few words that, as a disclaimer, it's intermediate results. So we trained the simple models to see what are the trade-offs, how much accuracy you lose if you try to protect your patient inputs in order to not to know it. As a data set, we took GTX dataset that is public, quite popular data set with 53 tissues and more than 800 people. As you can see on this violin plots, it's quite imbalanced. And it also has one problem, that they round it as not as good as if you would have proper aging values. So we trained the following models. So in the middle, you can see classical centralized models when you have access to all the data and you train either neural network or you train decision tree gradient boosting model. And we also, on the bottom, we also tried to simulate 4 private kind of hospitals that have this data, and you train on it and get some results with federated learning. And on the top, you can see that we also tried to make some generative models, kind of generation, and teach some classifiers on not private data, but generated data. So a few words about centralized results. So on the left, you can see the genes that were selected with Shapley additive explanations. There we applied 2 models, and surprisingly, light GBM gradient boosting decision tree model performed better than neural network, and it was somewhat more robust. So we used it as a standard of the art, and we compared it with first synthetic data that is generated with variational autoencoders. So in this like theoretical explanation, you can see that you have some parameters. Here it's just some facial features, but in our case it was age. So you can generate a gene expression for particular tissue, for particular age. And the idea is that you train a generative model on private data to which you have access, and then you make public synthetic data, and whole world can train on synthetic data. So here is a quite simple architecture of our variational autoencoder. Here is reconstruction loss, and the second technology that we compared with centralized was federated learning. For this, we use famous PyGrid library. So federated learning is what you all experience when you work with your mobile devices, because type suggestions and other services are actually machine learning models trained in a federated manner when they don't send all the data all the time to central server, but they try to train it on device and send model updates. In biomedical setting, you can actually see that it can be quite painful in terms of paperwork and privacy regulation to just give data from a hospital to a third party. And if the data cannot go, like in the picture in the center, to the model, to some centralized location, the model can go to the data. So we have some centralized model, we send it to all the hospitals. Inside the hospitals, they train on their own machines without giving away any data, those models, and then they return back not the data stays in place, but updates of the global model that are merged, and you have model 2.0, you again send it to all hospitals, and you have a second, third, and more federated rounds. We had the Google Summer of Code project to actually research it a little bit, and we played with Pi network that is like a way how you can dynamically make connections between data scientists / model owners with data owners who can be hospitals or companies, and how you can dynamically organized federated learning. So our experience, unfortunately, was mostly about dealing with extremely popular, but quite immature libraries. And we spend most on time of it instead of tuning the models. And as I mentioned, here is actually comparisons of all the models. So as expected, centralized models, they perform the best with R-square about 0.6. If it would be not rounded data, it would be better. Or if we would spend more time on tuning, for large GBM, we could even decrease number of features. For synthetic models, we actually didn't manage to train an external model only based on synthetic data. The results were quite poor, but we figured out that you can at least substitute half of the data. Maybe half of the data is private and half of the data is public. So you substitute half of the data with synthetic data and you get pretty decent results. For federated learning, actually, hyperparameter optimization and is quite complex because you can have different ways how the data can be imbalanced and distributed. So overall, you often get 2 times poorer results, unfortunately, but we are still tuning it. And there are sometimes problems with convergence. So after doing these comparisons, we came to some conclusions. So we see that there is still a huge potential to use a lot of data from other diseases and have distributed a network of models and data sets. Unfortunately, accuracy is a price to fit with the regulation and to do everything private. And we probably need to wait 1-2 years when we can train stuff with real patients in a totally secure way and with proper accuracy. And also we cannot just use generative model everywhere yet. And our next steps actually will be testing 2 other privacy preserving technologies like homomorphic encryption and secure multi-party computation. And we have to put a lot of time actually on improving the models and seeing how we can train better. With this, I want to thank you for your attention and ready to answer your questions.
Thank you very much, Anton. David asked, Anton, can you read for yourself or you want me to?
Okay, let me just check. Maybe you can read because I'm
okay. And in all the talk yesterday on Godfellow, the originator of the AI concept of GANs, the generative adversarial networks, talked about using GAN for differentially private data generation. This matches column 1 of your comparison. What problems do you see with it? It was also stated that in the webinar that knowledge about GANs has...
I know. So I kind of thinking about taking this Coursera specialization. So there is a specialization on GANs by Andrew Ng. So the thing is that we haven't tested the GAN here yet, but we plan to. So GANs, they from one side there are a bit, you can get a bit better results than with variational autoencoders. From another side, as you have like a sword and shield fight, it's harder to train them. So it can be hard to actually get results that are very, very close to real data. And from what we see now, probably until there will be some breakthroughs, we would think that they would be used like data augmentations, like you can say train some GANs on private data and you mix it with public data and you get better or comparable results. I think that's probably the use case for the next 1 or 2 years, sorry. But of course, things can get better. Any other questions?
That's it. Thank you very much, Anton. Exactly on time. The next speaker is Alex Zhavrankov from In Silico Medicine, who will speak on deep biomarkers of aging and longevity. Please, Alex.
Sure. Thanks so much. Can you see me? Because yeah. Right. And you can see me, and I'm going to be sharing my screen. Can you see my screen?
But maybe also make it full screen. Now it's still slide view.
Okay, just a second. And I need to do it like this. All right, guys. So for those of you who do not know me, my name is Alex Javankov. I'm the CEO of In Silico Medicine. We focus on drug discovery. I'm the Chief Longevity Officer of the company called Deep Longevity, which was recently acquired by another company called Region Pacific, and is publicly traded in Hong Kong, where I'm calling from. So it's almost 2 a.m. my time. Since it's a public company, I need to make this disclaimer, so it's not an investment advice. It's publicly traded, so don't buy the stock based on this lecture. The technology that I'm specializing in is called deep generative reinforcement learning. So Anton, I've noticed, covered a little bit GANs and variational autoencoders and generation of synthetic data. So we've been doing this for quite some time, almost since the very dawn of the deep learning revolution. In Silico, we use this technology for biomarker development, biological target identification, molecular screening, molecular generation, clinical trial analysis, and at Deep Longevity, we do biological age prediction, also biomarker development, biological target identification, synthetic data generation, real-world data analytics, and now we even got into motivation, behavioral modification, mental health, and depression. So publications are coming up. Actually, in this area, the many features that we work with are highly interpretable, so it's very easy to interpret with human mind. The history of deep learning, I'm pretty sure most of you are not familiar with that. Neural nets, the concepts of neural nets date back to 1940s, and many of the concepts are not new, but GANs, the topic that was previously touched on, are reasonably new. So Ian Goodfellow introduced them in 2014, and in 2015 we met Ian, and I started working quite actively in this area, and we use it very, very often in molecular generation. So we published our first paper in generation of novel molecules using GANs in 2016. It's the first peer-reviewed paper in this area. And then we did many, many papers lately showing experimental validation where we synthesize the molecules. But we also use it to generate synthetic human data. And a very good introduction to GANs and specific application of GANs was this paper by Reed in 2016, published approximately at the same time with ICML when we submitted our first paper on molecules, and approximately at the same time we did our first age predictor using deep neural networks. So you have two networks, one is a generator, another one is a discriminator. Generator is generating meaningful output, meaningful noise in response to textual input or some other generation conditions. And the discriminator is capturing whatever is coming out of the generator and comparing it to ground truth. And those two networks compete with each other. One is trying to trick the discriminator. The discriminator is trying to catch the generator generating fakes. So when they're trained, you can get this kind of images, again, 2016, so poor resolution. You can get this kind of input. This small bird has a pink breasting crown and black primaries and secondaries. And you can see that the direction of those generation conditions is there. Those are not Google search, these are generated. So GANs are great at generating pictures. Again, those people do not exist. So this is synthetic data, again, previously touched on, but that's very old stuff, so 2017. And deep neural networks are great at predicting age. You can see that it can predict age based on synthetic data. This kind of brings us to the concept of deep aging clocks. You can build aging clocks and predictors of age reasonably easy with shallow models. So you have just one input layer to predict age. Deep models allow you to have many, many neurons on the input. as many as you have input features, and one neuron on the output predicting age, with many layers of neurons in between in a basic feed-forward network. This is called latent space, hidden layers. And as this network gets exposed to data, to a lot of data, it learns to predict age with high accuracy. And it can tolerate quite a bit of noise in this data. So if your data isn't perfect, but it's real world data, the deep neural network is great at generalizing. So you can embrace dirty data, but you really need to have a very good test set. And we realized that quite early. So we started using large populational data sets, longitudinal data sets on many, many data types. So we built the first transcriptomic aging clock, proteomic aging clock, metabolomic aging clock using deep learning. We have pictures, EEG, CG activity, blood tests, behavior aging clocks. And you can combine those data types now using deep neural nets, either by building ensembles of deep neural nets or training on multimodal data. And then you can derive pathways, targets, causal graphs, and generate synthetic data using those deep neural networks. But this, in 2016, when we first published, that was a pretty novel concept. And now quite a bit of our work is related to how to make this concept more explainable and more and how do you derive causality from those deep neural networks. One really cool idea that we came up with also in 2016 was to train on age, where a lot of data is available. So everybody has age. That's the beauty of aging. To train deep neural networks, and there is a lot of publicly available data. So you can train deep neural networks on many of those data types to predict age, and then you retrain them to predict the health status, to predict disease. And then you can look at which features change from age to disease to be able to identify the features that are more disease specific rather than age specific. And you can do it within population groups. So again, my group is doing quite a bit of this kind of research. And if you look at the deep biomarkers of aging in history, there are many published in 2016, 2017. So again, we were the first to publish on hematological aging clocks, got reasonable accuracy from 5 to 7 years. There were tissue-specific transcriptomic clocks. Our accuracy is approximately 6 years, but can go up all the way to we can go down all the way to 4 years when we have enough data. We also published the microbiome aging clock, about 4-year accurate. There were also aging clocks published on X-rays, on MRI. You can do it with facial images and many, many other omics data. We published a clock with a company called Haut.ai, with Anastasia Georgievskaya, on facial images. When you have really high resolution images and really large data sets, high quality data sets, mean absolute error goes down to 2.3 years. It's pretty much almost as good as methylation clock. And again, there are many, many clocks. We try to have an integrated view on those clocks and combine those clocks in a systematic manner, either training on multiple data types at the same time or training within the data type and combining those predictors together in a weighted average fashion. And currently we have even behavior aging clocks, psychological aging clocks, clocks that are based on medical history, so with very interpretable features. And working with those kind of data types allow you to do very advanced feature engineering that will later help you to work with biological data. So you try to find features that are not directly correlated with biological age, with chronological age. Like for example, age of your spouse or age of your children. You don't want to have those in the features that you're training on, because that's very correlated with your kind of with your place in lifespan. So it means that you are old enough to have children, and they're already kind of, and you have them, and they are of specific age, or age of your spouse is very often correlated with your chronological age. So you don't want to have those, and you want to have features that are highly modifiable. So, like for example, features that age of death of your parents is something that you cannot really change. You don't want to have that. Again, learning from easy to interpret data types is something very important, and that helps you define the criteria for feature engineering for other clocks. We have quite a bit of papers supporting those clocks, all the way since 2015, 2016. So I'm going to cover some of those, and Polina Momoshina, who is going to follow with a presentation on more kind of feature importance analysis. We did the hematological aging clock. We showed that it works in smokers. So we can show that smokers are predicted to be older than non-smokers. We built a transcriptomic aging clock, microbionic aging clock, showed those clocks are population specific and also very relevant to mortality. So the first clock that we published was this deep biomarkers of human aging by Putin. So now I have, I think, 8 or 9 papers with Putin and PubMed. It's funny. This was the first application of deep neural networks on blood biochemistry data for age prediction. We also showed that now we can do permutation feature importance, so create a vector of all those features and randomly shuffle some to understand which ones contribute most to the accuracy of the predictor and build those beautiful diagrams and see which features are most important. So we've identified albumin, glucose, token phosphatase back then. And also showed that we can reach a pretty reasonable accuracy, so about 5.5 years, and ping some of those features to exact systems. And also back then, we realized that there is anecdotal evidence that people who look older than the chronological age to the deep neural net, they have various health conditions. And people who are younger are usually quite athletic and fit. I think I have to accelerate and skip some slides, but for our first study, we used approximately 62,000 samples from people who are more or less healthy and trained an ensemble of 21 neural nets, stacked them into an ensemble, and got to a pretty nice accuracy. So now it's by far much more advanced. We use a lot of generative systems and get to much better accuracy and can handle many populations. Also, we showed that many of those clocks are very responsive to lifestyle choices. So again, the first anecdotal evidence was coming from this first exercise in 2016. Then we showed that on large data sets and Canadian data sets for smokers and Chantix users, we showed that people who are smoking, they look much older to the deep neural net early in life, and actually many of those who are less genetically resilient to aging and other conditions, they die off, and by the time they're 7 younger, a little bit they're smoking. And we also showed for the first time that people who are significantly older than the chronological age, so 5 years or older, also have much higher hazard ratio. So, more than 2x chances of dying of any cause within any population if they're older than a chronological age, and vice versa if they're younger. So when you test those predictors on data sets that are also annotated with mortality. We also built a microbionic aging clock, kind of showing that this data might be useful for aging research. We built a predictor that is 4 to 6 years accurate. And we also showed that some species using feature importance and SHAP analysis, we showed that some of them are more kind of seno-negative or seno-positive. So some of them make you older or younger. We integrated many of those clocks into the systems developed by our company called Deep Longevity, which was acquired, and integrated those clocks into a centralized system, which is called AgeMetric, which kind of grows as you add additional data types into the system. So we can start with pretty much nothing, just asking you how old you feel and your chronological age. And then as you add more data, photo age, methylation aging, heart age, blood age, microbiome age, the system grows and creates a weighted average of those clocks. Now I have to accelerate because I am out of time. We provide a web-based system which allows you to track this age metric in time and individual features as well. And age metric reports to clinics that allow you to interpret those clocks in a pretty advanced manner. So you can now see which features make you younger or older, and which ones should you modify to look younger to the deep neural net. Not necessarily, again, it might not necessarily make you younger, but we have an algorithmic approach which allows you to see what you need to change to look younger to the deep neural network. For example, if you want to become 10 years younger, which features do you need to change in your blood? Various interventions, like for example, if iron levels is something that is making you look older, you might want to modulate your iron with iron supplementation, or same for cholesterol. So that can be modulated with statins or glucose or features. And we do the same thing on transcriptomics. And I'll skip a few things. Actually, we're to build an ecosystem around those clocks with hospitals, insurance companies, clinical providers. And I think that the idea of employing those techniques for life insurance is a very, very cool idea, because in life insurance, the interests of an insurance company and the individual are aligned. And so the individual wants to live longer and wants to, of course, ensure that there is a higher premium being paid at the end of life to their family. And the insurance company wants to postpone the payout of the premium. So they can provide additional aging clocks and look at what needs to be changed in order for the individual to live longer. Plus, it's a nice perk. So we are developing a system which is, costs you $150 to test how old you are biologically, using very, very primitive tests like blood tests, plus a lot of other behavioral and social data to be very predictive of mortality. And are collecting now large data sets. Of course, we also use federated learning in cases where it's needed, and collaborate with multiple clinics. So if you're a clinic, you can give us a call, and we would love to collaborate. If you're in China, you can add me on WeChat, and here is my email. I'd like to open the floor for questions. Thank you.
Thank you very much, Alex. I think we can still ask 2 questions. So I'll start with my own question as a moderator, something general that's been occupying me. How do you ascertain the reproducibility of the results? How do you ascertain that the results obtained with the algorithm on specific data set that was available to you will be also applicable in some other data sets that you've never seen?
So sure, to do that, you just need to test it on independent data sets and coming from different populations. So the speaker that is going to speak after me, Polina Mamoshina, who has been working with me for over 5 years now, you can look at some of her papers, and here, this paper, Population-Specific Biomarkers of Human Aging, a big data study of South Korean, Canadian, and Eastern European populations, published in Gerontology Section 8, really cool journal, is, I think, one of the first studies where we showed that we can train those deep neural networks within different populations, so Koreans, Canadians, Eastern Europeans, and then test Korean data on Canadian predictor, for example, to look at which features are different, which features are the same, how does the performance of one predictor compare it to another, and you can combine some of those predictors as well. And it was the first time when we showed that you can also test on data sets that contain mortality data within every population, and then on a combined predictor. and showed that that phenomenon that we've identified, that I talked about in one of the slides, that if you are 5 years or older in every population, your hazard ratio increases.
Thanks, and kind of combined question from David Wood and Joe Ferber. Basically, how sensitive is the prediction of age from facial photography? Is it influenced by makeup, mild sunburn, transit emotional state? And John Ferber adds, would it be influenced if somebody stayed up all night? What's your?
Yes, of course. So it's highly sensitive. And again, it kind of shows you the limitations and benefits of deep neural networks. If you were to Photoshop your face or apply makeup, the deep neural network would be confused and think that you are younger. And the best company that I know, and I'm an advisor to that company as well, is called Hout AI. Which does this kind of research on different photographic data types, either on very ultra high resolutions pictures of skin. So you don't even do it, you can do it on this macro, my micro pictures of your skin. And you can do it on entire face and see what features are making you younger or older. And then you can correct those features. You also see what those features are on the photograph, either on the full face or on a specific piece of the skin. And you would be able to influence some of those features using makeup. But of course, sleep and exercise and your body composition. If you have a lot of subcutaneous body fat, you will actually look younger because the wrinkles are corrected. And how your mood changes your age. What we also found is that if you just train on the population, if you smile and show your teeth, the deep neural network will recognize you as younger.
Thank you very much, Alex. And now to continue, Polina Mamoshina, who is also from in Silico Medicine, head of the biomarkers development at the company. She will speak about explainable deep aging clocks as models of hypothesis generation and aging research. Please, Polina, if you could make it shorter, we kind of.
I will try to be brief. Thank you. So I have to first make a disclaimer that I'm currently Chief Science Officer at Deep Longevity. It's a spin-off company of Silico Medicine, so I'm no longer part of Silico. And again, because we've been acquired by a public company, I have to make a disclaimer that it's not financial advice in any way, and you cannot use my slides as a financial advice. So a bit of a company. Again, just to recap, we focus on deep aging clocks, and my presentation will mainly cover those. And we've been founded by Alex Zhalenkov, as well as in Silico, who is also a long-term mentor of mine. So just to summarize what we have in the field in terms of markers of aging. So there are multiple of those that has been proposed since 2013. That was mainly driven by the accessibility of the data type in the public domain. At the same time, all of them, they were developed using standard shallow machine learning techniques. And now we are experiencing a revolution in algorithms. So there are new types of algorithms available, including the deep learning based algorithms, deep neural networks, and reinforcement learning algorithms, and they are the one who are generating most of the hype around AI, but they are also the one who are generating the credible advances. And now we believe there is a revolution in deep learning in healthcare area. So why it's happening right now, why most of those ideas came from 1980, it's mainly because we have advanced algorithms that we can use to train them on. Before it was only theory. We cannot actually train them. We also have GPU computing. It's only possible to train them with GPU computing and big data, so millions of samples to train because they're exceptionally data hungry. And fortunately, regulatory already recognized that, and since 2017 approved 14 purely AI-based tools. So many of them were based on convolutional neural nets, but also other deep neural networks used to develop those, ranging from the Arteris cardio MRI-based heart interpretation to one of the recent approval that Apple gained for their atrial fibrillation detection, which is actually now available for someone who is using the Apple Watch. And for most of those tools, FDA granted fast track approval. So we believe that having a deep learning as a type of algorithms used for biomarker development actually going to give us an advantage when the field will be ready talking to the regulatory. And since 2017, luckily, many of those deep learning based clocks has been developed. We as a team contributed a lot, but also other teams be using completely different data types to build them. And apparently we end up in a situation that there are a lot of aging clocks proposed. So which one we actually use? So we believe that there is no universal marker. And if we analyze the data, even for people same chronological age, and that was a really nice study done by Professor Dan Belsky and published in 2018, when he and his team analyzed the people same chronological age, healthy people, using completely different markers of aging. And what he saw was interesting, that even between different methylation clocks, there was little or no correlation. While if we analyze the longitudinal data, we see that clocks actually ticking in the right direction. So people without interventions, they getting older, not only chronologically, but also biologically. So they're working and they just measure different processes. That's why in a company, we focused on a really diverse portfolio of clocks, ranging from really simple data types like for like facial images to really complex transcriptome data. Again, we proposed some of those clocks already, we published them. One of the recent one that we published was for microbiome, when we showed that not only we can actually predict age of healthy individuals with their whole genome sequence profile of microbiome in their gut, but also we can see that the individuals with type 1 diabetes actually predicted older by their microbiome. So they show signs of accelerated aging according to the microbiome. This model was somehow accurate, but what is really interesting to do with this model is actually that because it's a computation model, we can explore how the changes in input parameter would affect the output. In this way, we can actually design those trajectories for each of the species in the data to see which of them actually promote useful state and which of them actually promoting the negative state. I mean, causing acceleration of age to the model, and that's how we're able to classify them, but send negative, send a positive status. What was really interesting also to understand is that once we identify where the individual is on those trajectories, we can actually suggest them in which direction they should move, they should shift their microbiome profile to get younger, at least for a deep neural network. While with microbiome, it's a bit tricky because it's not like we have a lot of interventions with known actions on the species. At the same time, we had other data types that we can actually experiment with. And those are clocks that are based on clinical blood tests. Again, if we look at the data itself, blood markers are not markers of aging by themselves. Of course, we see trends there, like for example, albumin tend to decrease with age, cholesterol tend to increase with age, glucose tend to increase with age. At the same time, it's a bit hard to actually get age as a number from those values unless you have a model that actually capture the non-linearities between changes in blood marker levels and age. And we did it with an ensemble of deep neural networks. And now we also show that not only we can predict age of healthy individuals rather accurately with a simple blood test, but also that smokers are predicted older, twice as old compared to non-smoking peers, that the patients who are predicted older, they have higher 5-year mortality rates and lower life expectancy and other way around. But what other things we can do, again, because there are computation models, we can actually design those trajectories the same way we do this for microbiome, for blood. And this is where it's getting really interesting, because for blood, we actually know what kind of interventions we can suggest to shift blood profiles. And by doing this, we believe that we are introducing the optimal blood levels rather than a reference one. Reference ranges, of course, are really important when we define the pathology state, but if we think of wellness and the optimal performance, we actually have to focus on optimal blood parameters. And here's an example for one of the analysis that we did for this particular individual. She can actually got at least 5 years younger by shifting their blood parameters, and at the same time only total cholesterol was out of range, was one of those parameters that was abnormally high and recommended to change if she would go to a standard care practitioner. So we believe those are really powerful tools to do those kind of simulations. So just to summarize, again, there are multiple aging clocks available, and these conferences are also one of those examples. A lot of people presented completely different approaches to biomarker development, but we believe that using AI and deep neural networks in particular, we can actually design our next generation aging clocks. And they are not only useful for age prediction, but also for identifying those variables that are associated with age the most. So ultimately, because they're computational tools, we can actually use them to simulate the effect of those changes in the input parameters on the outcome, and therefore even like simulate interventions. So we believe that those deep aging clocks can be the one that can be used to run so-called in silico clinical trials, and therefore will be really useful in clinical trials in general. And on a side note, some of those clocks that I showed you, and again, algorithms that are optimizing blood parameters, they are integrated into our Young AI web platform, and it's live now. So we welcome early adopters to register there and provide us with feedback. It's totally free now for early adopters, so you can try it on, you can try it with your own data. So thanks for your attention, and I'm open. I hope I have some time for questions.
Thank you very much. One question from Phil. Other issues depending on the chronological age of the patient versus available data. For example, is it possible to be accurate from age 18 to 80? In addition, might there be more emphasis on life-threatening biomarkers?
Right, so there is, it's a good question. So there is a common problem for those kind of models called linear fit problem, that usually on the tails of distribution, the model is less accurate. And it happened to be the case that for most of the data sets out there, the distribution for ages is normal. It's meaning that you have a median for, I don't know, 40, 50 years old, and most of the samples out there for them, but for people who are over 80s, there are less samples available, so the model tend to mispredict them. At the same time, there are ways to correct those predictions for the tails of distribution. So there are tricks there that you can computationally apply to avoid any problems with those people. But in general, we should also expect people who are over 80 or 90 to be a bit biologically younger compared to others, because it's already a rather long lifespan if we're just thinking of like average lifespan. I mean, depending on the country, but still.
Thank you very much, Paulina. I move to the next speaker, Sven Batagis from Achilles. Will speak just about biomarkers of aging.
Okay, you can see my screen, I guess. Okay, so well, thanks to the organizers, I will say, but I was one of the organizers. So welcome everyone. I'm going to talk a little bit today about biomarkers. Oops. Why am I not? Okay. Yeah, so I work at the Brakman Lab in Ghent University, but my research is a collaboration with the biotech company Rejuvenate Biomed, and we are funded by the Agency of Innovation and Entrepreneurship. So I should disclose first that my views expressed in this presentation are my own, and they do not reflect necessarily the views of the company. So first I'm going to talk about actually the thing I'm specialized in, and that's how to discover new drugs on aging. I was asked to talk about biomarkers, which I admit is not my field, but I'm going to do a trial here to talk about them. I'm going to talk first about what biomarkers are. I'm going to look a little bit at how the field is evolving, and then I'm going to look at one specific biomarker of aging, and then come to a conclusion. So how do you discover new drugs involved in aging? Well, traditionally, we can say there are two very main big paradigms. On the one hand, you have the phenotypic screening, and on the other hand, you have the target-based screening. Now, there are other paradigms as well, such as in silico screening, where you could, for example, look at the differences in the gene expression pattern between young and old and see if there is a compound that shifts the gene expression profile more towards that of a young cell. But mainly I'm going to just focus on these two. So in phenotypic screening, that's the classical way in how you do drug screening. That was used 100 years already ago to discover the first drugs in humans. And so the mice has always been kind of the organism in which most of this work was done. But these days, phenotypic screening is mostly happening in cells, and also work on aging has been done in cells, but not that many screenings. One example was a recent screening by Cynthia Kenyon, where they looked at 100,000 compounds in cells, and they looked for compounds that increased oxidative stress resistance. They found 32 main hits that were then validated in C. elegans, and they found 9 compounds that extended the lifespan in C. elegans by between 10 and 50%. Also, of course, analytics have been checked and found through screenings in cells. C. elegans has been really one of those organisms that is very strongly upcoming, and as I will show, actually it is the most used organism for drug screening in geroprotective drugs. But also target-based approaches have been used, not that often, but a few of them exist. For example, of course, we probably all know about the SIRT2-in-1 activating compounds that were discovered using target-based screenings, but also, for example, inhibitors of CD38 have been discovered. And when one of those compounds that GSK developed was given to mice, it was shown to improve the health of elderly mice, including an improvement of glucose tolerance, muscle function, and cardiac function. So when we look at the drug age database, we can see that there are almost 600 unique compounds that have been screened that are in the database. By the way, the database is probably not complete, but it's almost complete, I will say. And about 70% of all the compounds in the database have been screened in C. elegans. The second most popular model is the fruit fly with about 23%, followed by the mice with 10%. By the way, this doesn't add up to 100% because, of course, some compounds have been screened in more than one organism, luckily. However, not that many compounds, surprisingly. So I have, in analogy with genetics, where you talk about forward and reverse genetics, I have called it forward screening and reverse screening. Forward screening being you start with C. elegans, Drosophila cells, and when you find hits, you then move them to the mice. From the mice, you move them to dogs, to primates, for example, and finally, of course, the goal is to move them to humans. Reverse screening is when you have hits in higher organisms, and that has only been in mice, and you then move them to simpler models. Now, surprisingly, when you look to the drug age database, there are very few drugs that you can find that were discovered in C. elegans and Drosophila, and were then moved to the mice. So these three compounds are the ones that I found. There might be a few more, but not many. In many more cases, you actually see the opposite. Drugs were first discovered in the mice, and a nice example here is metformin, which was discovered in the 1980s by Dillman and Anisimov. Well, back then it was fenformin actually that they first discovered, but later they also screened metformin and buformin in mice, and it was found to extend lifespan. Yet it took until 2002 before buformin was first tested in C. elegans, and actually 2010 before metformin was finally screened in C. elegans. So this is examples of reverse screening. So that ends the drug discovery part. I'm going to move on to the biomarkers. So there are many different types of biomarkers. You have, for example, safety biomarkers, liver enzymes, for example. When you screen new drugs, you want to see if those drugs do not cause toxicity to the liver. You can do that by monitor liver enzymes. You have also prognostic biomarkers, for example, that will allow you to predict the clinical course of a disease, as well as diagnostic biomarkers that allow you to diagnose a disease. But the things that we are interested in here are the surrogate biomarkers, the one that actually can be used as endpoints of clinical trials to show that an intervention actually works in humans. So what are a good criteria for a biomarker of aging? There was this paper 2 years ago in Euroscience where these 4 criteria were proposed. The American Federation of Aging Research has proposed 4 other criteria, but they largely overlap with these criteria. So first of all, what you want is that you have something that you can measure relatively cheaply, easily, non-invasively in humans, that you can measure more than once in humans. So it doesn't require like tissue samples through biopsies or things like that. So often blood-based will be ideal for this. But a measurement must also be reliable, and it must also be very reproducible. Then the second thing you want is that it's very relevant to aging, obviously, and also that specific diseases that are not related to aging do not disturb this biomarker too much, so that there are no specific diseases where the biomarker suddenly goes haywire. A third thing you want is that it's very robust and consistent in its ability to predict all-cause mortality, as well as clinical and functional outcomes. And finally, you want, of course, if you are going to use this as a validated endpoint in a clinical trial, it must actually be able to not only show responsiveness to an intervention, but actually predict that this intervention then is going to extend lifespan. So the biomarker field is really booming. And so here you can see in orange the total number of papers in PubMed, and it's growing obviously, but here in blue you see the number of new biomarker papers, and it's actually growing at a much faster rate than the total number of papers in PubMed, showing that there is actually quite a lot of interest in biomarkers in general. When we look at biomarkers of aging, and so I just screened PubMed with the term aging, both in British and American English, and biomarker, then this is what you find. Of course, this is missing quite a few papers, so this is definitely not an exhaustive search because it ignores everything where the search term will be biomarker of aging. That will not be included here. but you can anyway see that the number of papers on aging biomarkers is really increasing over time, and it has really exploded in recent years. And when you then look at all these papers individually, you find quite a lot of different biomarkers in them that have been proposed. Some of them have been discussed, of course, today, like DNA methylation and the photo-age clock, for example. But the one that I would like to talk about is the N-glycans. And the reason I want to talk about that one is because it's probably the one that you might not have heard about. It's not a very popular or well-known one. So what is N-glycosylation? It's a code translational modification that happens in the endoplasmatic reticulum. It's when this mannose structure is attached to an asparagine residue of a protein. And about 50% of proteins have these types of glycosylation modifications, particularly proteins that are secreted by the cell or proteins that end up in the cell membrane. So after being modified in the endoplasmatic reticulum, it gets translocated to the Golgi, where a lot more modifications happen to this glycan structure. And then, of course, from the Golgi, it goes to where it needs to be. So how do you measure them? There are many methods out there based on HPLC, based on mass spectrometry. However, I decided here to stick myself to one method that is actually a method that is very easy to do and that doesn't require complex equipment. And this method only requires 2 microliters of blood, so very small sample size. So you take a little bit of blood, you cut off first all the proteins, so that ends up with 3 glycans. You label them with this fluorescent dye. Then you remove the sialic acid groups. The reason is they are negatively charged, so they will interfere with your separation. You only want a separation based on this fluorescent dye, which has 3 negative charges to it, as you can see here. And then you use a capillary electrophoresis DNA sequencer, so the classical Sanger sequence type machine, to actually separate these, and then you end up with a chromatogram of different glycan peaks. Now, this is not the most sensitive method compared to HPLC methods, but it is a method that is very easy and available to a lot of labs. Lots of labs have these Sanger sequencers. And so it's feasible to do in a bigger clinical trial. So what you then see if you compare young with old is mainly actually a change in peak number 1 and peak number 6. And these are the 2 glycans that correspond with peak number 1 and peak number 6. They are both B-antenary glycans. And so you can see they are also related to each other, namely that this one has actually the galactosidase removed from the end of them. And when you then take the ratio of these 2, peak 1 divided by peak 6, and you take the logarithm of that, you end up with what is being called the glyco-age test. So why are they interesting to use? As I already mentioned, it can be done in a very small sample size of 2 microliters. It can be done both in plasma and serum. It can be done on frozen samples, and you need very little equipment. So that means it fulfills the first criterion. It's also very robust. So when you repeat the measurement, it stays stable. It doesn't have a lot of technical variation. The second criteria, is it linked to aging? Yes. So here you see data from the Chinese, the Belgian, and the Italian population. And you see in the Chinese population, both in males and females, an increase in peak 1, an increase in peak 2. And in females, you see a very strong decrease in peak 6, but not in males. And then in the Belgian and the Italian population, when you make this ratio of peak 1 and to peak 6, you see it increasing all the way up to centenarians. I should mention that it stays relatively constant until you are in your 40s to 50s, and it's only thereafter that you really see it increasing. So then is it also linked to clinical outcomes? Yes. So if you look at people with dementia, they have higher glyco-age scores than similar aged controls who are healthy. And when you look at people who have Cockeye syndrome, which is a defect in transcription related nucleotide exarch repair, you actually see that those people have much higher glyco-age test scores compared to the healthy controls. Even they score as high as people in their 90s. So can interventions do anything about it? There is very little data in the literature about this at the moment. There is one unpublished mice study on calorie restriction that apparently shows it does. This is another study on calorie restriction. Sadly, in this study, the only compare that they did is CR compared with a medium fat diet, so not a real control diet. But you can see that there was a significant decrease in the calorie restriction group in peak 1. And as you remember, peak 1 is a peak that actually increases with age compared to the animals that had a medium fat diet. And you actually can see here, if you keep animals on CR and you switch them to a medium fat diet for 4 months, then actually it falls in between these two, but that didn't reach significance. Peak 6 is maybe a little bit surprising because with age, at least in females, in human females, it decreases. And actually here CR decreases it further. So that kind of suggests that peak 1 is actually a much better measurement of aging than peak number 6. So what are the remaining questions in this field? First, do interventions other than calorie restriction slow down aging? That slow down aging decreases glyco-age. Is the glyco-age accelerated in other progeroid diseases and just not only in Cockeye syndrome? Does the glyco-age correlate with aging in cell types that do not secrete significant quantities of glycoproteins, because most of them are secreted by the liver or by the B cells? A lot of antibodies, IgGs, are strongly glycosylated. But your kidneys, for example, or your brain will not secrete glycoproteins in secretion, in the bloodstream, I mean. So can the glyco-age test actually measure aging of these organ systems? Of course, the fact that you see an increase in glyco-age in people with dementia suggests that that might be the case, but it needs further confirmation. Are there other disease conditions that could actually interfere with the measurements of glyco-age is unknown? And can it be used in species other than humans or in mice, obviously? So let's conclude. It's a promising yet understudied biomarker of aging. It fulfills criteria number 1, for sure. It probably also meets criteria number 2. And the other 2 criteria, they are, well, maybe met. And they need a lot more evidence to really validate them. And so the endpoint is to go to validated surrogate endpoints for these longevity therapeutics. The problem, of course, remains the validation of the surrogate, which has to be really strongly validated. And that's going to be difficult. And we can also conclude that not a single biomarker will measure biological age across all the tissues. So you will have to go to composite biomarkers. And for example, I just took a random recent paper that talked about a meta clock for based on epigenetic clocks. So, but there are other papers out there that develop other composite biomarkers. So thank you, and I will take questions.
Then there's one question, I think, from Rakesh. How many DNA methylation biomarker companies are currently there? And is there a gold standard biomarker, in your opinion?
Well, I don't know how many biomarker companies there are out there, to be honest. Is there a gold biomarker? No, not yet. But I think that, of course, the methylation clock is probably at the moment one of the most advanced ones. But also functional measurements, right? Like hand grip strength and things like that really do correlate very well with mortality. So I think they are the ones that are the furthest, but there are many upcoming ones that are very interesting.
Another question from Aubrey: how are glyco-age and glycan-age related?
Oh, so they are. It's interesting because I didn't mention glycan age, but it's another one by another research group. They didn't use peak number 6, I think, for correction. They looked at some others, but I don't recall exactly which glycan structures they looked at, but they were just a few other glycan structures.
Thank you, Sven. We go to our next speaker, Alexei Moskalov from the Russian Academy of Sciences, I think. And he will talk also on biomarkers of human aging.
Dear colleagues, dear organizers, thanks a lot for the possibility to join this exciting meeting. Already mentioned that many physiological functions decrease with aging, that functions related to lung capacity, liver, kidney function, brain, and also skeletal muscle functions. Less known that not only decline of functions reflects aging, but the levels of some functions increase, for example, stress hormone levels. And to study aging, to understand aging, we need biomarkers, some measurable parameters that are reproducibly change with aging of a person. It will help us to predict the accelerated or delayed aging of an individual, to monitor the effectiveness of therapies aimed at preventing aging, including already mentioned diets, lifestyle, geroprotectors, physical activity, etc. Sven already enumerated some biomarker criteria. Most of the most important one, correlation with the biological age of the person, and also prediction of mortality better than chronological age, and the prediction of early stages of specific age-related diseases or multimorbidity. And also biomarkers need to be minimal invasive. I think we need some additional criteria to go to practice for translation. It should be sensitive to early signs of aging, because some kinds of aging biomarkers like frailty index very close to mortality, but not reflect early stages of aging, the stage where we can reverse some changes. And also have a predictive function in the foreseeable period, not in a very long period of life. And have a low analytic variability and a very high reproducibility. It is very obvious that it is difficult to select any single indicator as a biomarker of aging. We need a panel of biomarkers, or I think it's a very important reflection of what's the difference between chronological and biological age. Biological age of persons of the same chronological age are very variable. And also you can change your biological age by interventions. I use this classification of types of biological age, clinical and functional tests, such as anthropological markers like BMI, waist, hematological biomarkers, markers of inflammation, insulin resistance, lipid profile, endocrinological markers. Also, aging mechanism-based markers, because we know some about the mechanisms of aging, like telomere attrition, lysosomal or mitochondrial dysfunction, cellular senescence, and we can measure these particular processes to understand the biological age of the person. And also omics. If we propose that we don't know anything about the real mechanisms of aging, we can collect big data like metabolomics, transcriptomics, metagenomics, proteomics, methylation of DNA, or also multi-omics to predict a biological age of the person. I would like to discuss shortly the advantages and disadvantages of these kind of markers of biological age. For example, functional markers have a direct clinical interpretation and very useful for doctors, but even if they predict mortality better than passport age, it is unclear to what extent they measure biological aging itself, rather than health deterioration for other reasons. In addition, these markers often only work as averages over very large samples, for example, from wearable devices, etc., and vary greatly from one person to another person. Molecular markers are based on deeper understanding of the molecular and cellular causes of aging, and should theoretically be more sensitive to early signs of aging, as opposed to mortality and frailty. But like clinical markers, they lack stability and reproducibility for specific patients. This is because aging is a multi-level process. Therefore, markers of individual mechanisms cannot cover all its aspects. Omics, changes in omics are the results of alterations in the body at different levels, making them a useful way to approach the complexity of the aging process. But we need a reliable, harmonized data of omics of many people to make some such models. But multi-omics approaches to assessing the aging rate also take place. We recently published a review paper on multi-omics approaches to human biological age estimation in mechanisms of aging and development. You can look at this paper to go further. In collaboration with the gerontological clinic of Moscow Medical Institution, we looked at collected data of many patients of different ages, which were studied by 86 different parameters of their health, including many hematological parameters, endocrinological, inflammatory, some molecular markers like telomere length, and functional tests. And we applied machine learning to this data and observed that arterial aging parameters, 4 arterial aging parameters overperformed all other parameters in this study. And we established arterial index model and observed that it better predicts biological age in patients with diabetes and hypertension. And now we try to use this model in several small clinical trials. Very good parameter of biological age is frailty index. it's very, very easy to apply from different kinds of medical data. In collaboration with Arnold Mitnitsky from Canada, we applied two kinds of biological age estimations, frailty index and one of the AI bioage approach to COVID patients. And we predicted the risk of death using age, passport age, and also frailty index and AI bioage using this data. And we observed that age is the most predictable risk estimation for this kind of patients. But frailty index and AI bioage also are good predictors of mortality in COVID patients. We combined age, passport age, and the frailty index, and obtained more precise risk prediction for mortality in this kind of patients. It's unpublished. I also recommend you to look at our two books published in academic press, Epigenetics of Aging and Longevity, including epigenetic logs, and biomarkers of human aging, published in Springer. And also, if you need something less theoretical and more practical, you can join as a tester to our new project, Young AI. You can download the application for your iPhone or go to website to register to this project and learn your biological age with us. Thank you for your attention.
A question from Walter. Alexei, it seems like the attempt to measure biological age is doomed to limited accuracy because it essentially averages a bunch of factors. Some parameters might be in a critical stage and others not. Some might be treated and greatly extended the period of healthy life. Anyone that goes over the age can spell death. Trying to lump them together seems inherently too simplistic. What am I missing? So,
thank you for your notion. It's not a question. I think we need a real big data and multi-omics approaches to deeper understanding of biological age. And we will do it, I think.
Okay, thank you, Alexei. So our last speaker is João Pedro de Miguel from Liverpool University, who will speak about molecular signatures to decipher aging.
Hi, Elia. Thank you very much. So I'll just let me start sharing my my slides, and I'll start by saying, of course, thank you to the organizers for the invitation and for organizing yet another conference. It's a shame that we can't be in Brussels. Can you see my slides well? Okay, great. So I guess in the topic of biomarkers, most of the work we've done is at the level of transcriptomics. So why transcriptomics? I mean, if you think about it, really proteins are the actors in biology, as James Watson put it so eloquently. So why focus on transcripts? You know, we have proteomics, we have epigenomics. Well, the advantage of transcriptomics, most of all, is that it's relatively fast and cheap and can be done on a genome-wide, high-throughput fashion. So proteomics it's a much more cumbersome process. Now, of course, we've heard already about epigenetics. We know that the best biomarkers of age are Horvath clock and methylation clocks. On the other hand, the problem of them is that they don't really give biological insights into the processes that are changing. By the other hand, gene expression profiling and proteomics may not be as accurate biomarkers, but on the other hand, they provide biological insights. And they can, for example, for testing intervention, they can indicate what is changing in a given intervention or trial or whatever it's being tested. So, I mean, I'll just give a couple of examples from our own work. I guess several years ago, I'll start from several years ago, we did this meta-analysis of aging microarray studies, combining several different studies in mammals from different tissues. And the advantage of that, again, without going into details, that when you combine all of this data, that this eliminates the problems of each individual studies and improves the signal to noise ratio. So we can identify genes and pathways that individual studies miss. And so that is an approach that we've used for various other processes since then. So this was something we've done for aging, and we have, we've done for other interventions. So let me show you a couple of examples. And again, I won't go into a lot of methodological details, but happy to answer questions, or feel free to email me later. So we've done an approach as well related to caloric restriction, where we looked at this conserved gene expression profiles of caloric restriction across mammalian tissues and organisms. So various different species and different tissues. And again, we find this conserved signatures of caloric restriction. And this also allows us, because it provides biological insights into processes like metabolic processes that are changing in the context of caloric restriction, then this allows us to, for example, identify which genes are regulating, or maybe candidates for regulating these changes, new biomarkers, and also new targets for drug discovery. So again, I'm not saying that this is a better biomarker for aging than, say, epigenetics. The argument is that this can provide biological insights and targets that are simply not possible with, for example, methylation. More recently, we did this meta-analysis of genes associated with cell senescence. So we combine multiple gene expression, both microarray and RNA-seq, gene expression profiles from senescent cells, so which you see here. So here on the left, you see young fibroblasts, and these are senescent fibroblasts. And this was something I did, many years ago when I was a PhD student. So we combine all of these gene expression profiles to derive a conserved gene expression signature of cell senescence, just like we did for caloric restriction and for aging. And again, without going into details, one of the things we did then was we looked into how these genes change in normal human tissues. So what you can see here is for genes that are underexpressed in cellular senescence and genes that are overexpressed in cellular senescence. And as you can see, there's not a very strong signal across all human tissues. So this is data from various humans to be upregulated during cell senescence. There are more tissues than expected by chance in which these genes are also upregulated. So what that suggests is that indeed you observed signatures of cell senescence in normal aging, in normal human aging tissues. I would point out, however, that there are some exceptions. I think the most striking exception is actually the uterus. The uterus is quite striking because you see actually the genes that tend to go up in cell senescence actually go down with age in the uterus, and genes that tend to be downregulated in cell senescence actually go up in the uterus in women, in old women. So the point is that you see signatures, gene expression signatures of cell senescence in human tissues, in some human tissues, in more human tissues than expected by chance, but not all human tissues. And there are even some tissues in which you see opposite patterns, like the uterus. So, I mean, I would argue that this suggests that indeed cell senescence is more prevalent in older tissues, but there are exceptions. We cannot say that cell senescence is present widespreadly in all age human tissues. I mean, and for more details, I mean, we recently published this paper in Genome Biology that goes into a lot more details on this approach and our results. Now, the other aspect, of course, is in addition to microarrays, is that there's a lot of data from RNA-seq. As I'm sure you're aware, the cost of sequencing have dropped dramatically in recent years. And thanks to a variety of machines, we can now sequence genomes much easier, and also transcriptomes. So I'll show you a couple of examples. I mean, we did this RNA-seq of aging tissues in rats, I mean, several years ago. But this was I think it was the first transcriptome sequence of normal aging in mammals. And it's a good example of the advantage of RNA-seq in terms that we detected a number of differentially expressed transcripts that did not map to any known genes, any known exons, what's called dark matter transcripts. And we even identified some new genes that are not yet on genome annotations. So that's a good example of that. Then a few years later, we did this RNA-seq of caloric restriction in the brains of rats. And we showed, I mean, again, without going to a lot of details, that you see a lot of neuroprotective genes like thyroidoxin that are overexpressed in caloric restriction. So they go up in the brain of rats under caloric restriction. We also show that caloric restriction prevents most aging-related changes. So you see, compared to normal aging signatures, gene expression signatures, you see that there's a amelioration of these patterns in animals exposed to caloric restriction. Not all patterns, but most of them. And the other thing that we thought was interesting, that there were a lot of changes in chromatin-related epigenetic regulators, which suggests, hints, or we can hypothesize that there are mechanisms related to epigenetics associated with caloric restriction responses. Now, more recently, the other thing we've done is, okay, so we have all of these signatures of aging, of caloric restriction. So we looked into compounds. So I'm sure you're aware there's databases of publicly available databases of gene expression signatures of chemical compounds and drugs. So we're looking into the overlap between gene expression signatures of caloric restriction and gene expression signatures of drugs, trying to find, can we find drugs that overlap with gene expression signatures of caloric restriction, which is in turn associated with longevity. So the answer is yes. I mean, to make a long story short, you know, we tested 5 compounds, 5 candidate compounds in worms, 4 of which extended lifespan, and they extended lifespan in normal worms, but not in worms under caloric restriction. So this shows that we can employ this network pharmacology approach to repurpose compounds for life extension. So we can identify compounds that have other clinical applications that extend lifespan in animal models, in this case, worms. I mean, I think the most interesting compound, something we're still working on, is allantoin. This is actually a marker of oxidative stress. And we are now studying its mechanisms and also studying other related compounds that extend lifespan in worms as well. So we have some unpublished data. So maybe hopefully in the next symposium, ideally in person in the beautiful city of Brussels, I can tell you more details about it. The point for now is that we can use these gene expression signatures to identify compounds that extend lifespan, and that in this specific case act as caloric restriction mimetics. So in summary, I hope I persuaded that really gene expression profile, I wouldn't say it's the best biomarker per se, but it is a cost-effective way of deriving biomarkers. Okay, they're not as accurate predicting biological age as methylation, but they're more insightful from a biological perspective. That's the big problem of methylation. And you know, the Horvath clock is that, yes, it's accurate, but nobody knows what it means from a biological level. And if you're trusting particular interventions, you want to have insights into that as well. If you're testing caloric restriction or metformin or any compound, you want to know whether that's ameliorating aging, but also what sort of biological effects it's having on an animal or a patient or a tissue. So that's the advantage of gene expression profile, is that it provides biological insights, not just biomarkers. So I've told you of various microarray meta-analyses that reveal signatures associated with aging. And actually, we have a paper currently submitted on a new gene expression profile of aging based on a meta-analysis combining microarray and RNA-seq. We've derived gene expression signatures of caloric restriction as well, and gene expression signatures of cell senescence. I've told you also of RNA-seq, which we apply to aging, various processes, and we apply to caloric restriction that associates epigenetic mechanisms in neuroprotection and aging. And now, lastly, I told you of how employing network pharmacology using gene expression signatures, and this can be used to reveal new longevity drugs by combining gene expression signatures as we used of caloric restriction or other longevity intervention with gene expression signatures of drugs. So with that, I just want to thank, again, thank you for your time and attention. And I want to, of course, thank my lab for all of the work that I've told you. And I guess I skipped a lot of details, but all of our preprints and reprints are available on our lab website. If you have any questions, feel free to contact me, and also thank our funders. And before we finish, I also want to point out this company, Centauri, that I'm also involved in. Well, recently, I'm now leading their scientific efforts that inclusively we have a project actually that called Aging Profiles, in which we want to analyze multiple omics associated with aging to try to understand about the process of aging and how to better intervene on it. And so I would suggest you check out the website of Centauri, and if anyone is looking for a job in computational systems biology approaches in the context of aging, please feel free to contact us. And again, thank you very much for your time and attention.
Much, Jomirov. I don't see any questions in the chat, so I guess this otherwise somebody wants to shout out the question. No, okay. Then I guess it's a conclusion of our session. It's been an intensive, highly enriching session. Thank you very much, all the participants, all the organizers. I hope we meet again soon. The next session will be in 5 minutes. It's been a 2-hour session. So in about 5-7 minutes, we reconvene to discuss more general and more social aspects of testing and of biomarkers development. So thanks again, and see you.
Thank you.
Seen any grim reapers or wretched spirits, we have found cause and effect, that our bodies are governed by the same physical laws than the rest of the universe. Aging is a technical problem, and every technical problem has a technical solution. And we've found technical solutions to many diseases over time. And we have a pretty good history of finding technical solutions to debilitating diseases in the past. The byproducts of solving disease is that we get to live happier, healthier, and longer, though not indefinitely. Solving some form of cancer or immunizing ourselves against the next strain of pernicious Ebola will only be temporary victories. There will always be more diseases around the corner. So it seems we need to focus on something more fundamental. Interestingly, there's been many recent studies of the effects of therapies on the health span and longevity of mice. A senolytics approach has been shown to reduce age-related dysfunction and extend remaining lifespan by 36% following being administered on old, aged mice. The Ocean Biotechnologies lifespan study is an interesting example. testing how p53 and p16 proteins and genes affect age-related dysfunction, as they are noteworthy markers for cancer in senescent cells. The lifespan study is over 6 months old now, using cohorts in which gene therapy is deployed against either p16, p53, or both p16 and p53, and there's a control group which are injected with placebo. The mice were 2 years, that is 104 weeks, old at the start of the experiment. The mice, which usually live only to 3 years old, were 2 years, that is 104 weeks, old at the start of the experiment. The p16 and p53 mice seem to be doing very well indeed, in comparison to their peers. Even though the trial is nowhere yet near finished, the results so far are quite impressive. A concern that people have about living longer lives is that if one were living, say, to 150 years, for most of the latter part of one's life, that would be unbearable, living in an old, sick body. But this is unlikely to be the case, as the reason one could reach such an age is the same reason one would be living in a healthy body until that age. In any case, if you decide that at some stage before reaching 150 years that you don't like life anymore, you can opt for physician-assisted dying or step in front of a truck. But the point is, with working rejuvenation treatments, chronologically older people need not be living in older, sick bodies, but instead living worthwhile, active, healthy lives. This is important not only from individual perspectives, but also on population levels. Let's take a look at some statistics. According to the World Health Organization, between 2015 and 2050, the proportion of the world's population over 60 years will nearly double from 12 to 22%. By 2020, the number of people aged 60 years and older will outnumber children younger than 5. The pace of population aging is much faster than in the past. In 2050, 80% of older people will be living in low and middle income countries. All countries face major challenges to ensure that their health and social systems are ready to make the most of this demographic shift. These stats put things into perspective. It's important to see that health in old age is desirable at population levels. Rejuvenation medicine, repairing the body's ability to cope with stresses, or practical reversal of the aging process, will end up being cheaper than traditional medicine based on general indefinite postponement of ill health on population levels, especially in the long run when rejuvenation therapy becomes efficient. It buys so much time to figure out what to do next, how to improve the therapies to address the next wave of issues for individuals as well as on population levels. So obviously, this will not only postpone ill health and expenses for individuals and populations, but also improve general quality of life for everyone. How valuable is the anticipated gain, not only for the individual, but for the world? Anticipated health gains over periods that would normally be dominated by ill health means extra productivity and more consumer spending. Then on top of that, the gains of longevity that keep paying dividends in the economic usefulness of expertise gained over long, healthy lives, and in the general social benefits of wisdom of individuals who have been around for a long time. So, back to one of the questions at the start, what might we do if we thought we could cure aging? Well, let's call it indefinite postponement of ill health associated with aging. The notion of ourselves being around for a lot longer might inspire us to be more concerned with longer-term issues and more readily act on things like climate change and maybe trying to avoid thermonuclear war and avoid other global catastrophic risks. So there's an obvious personal and societal financial disincentive to constantly battling the ailments that occur as a byproduct of a worn-out body. Instead, it will be cheaper to rejuvenate the bodies periodically into good health. If people's lives are extended, then it also makes sense to have their health span extended. The lifespan of humans has been steadily increasing over the past centuries. But there are reasons that simple extrapolation of past trends is not a great predictor of how lifespan may change in the future. What anti-aging seeks to ultimately achieve is to reverse fragility, rejuvenate the damage that contributes to sickness, preventing the body from getting frail in the first place. which will, as a side effect, reduce the amount of diseases the body is susceptible to, and also as a side effect, increase longevity. I wouldn't mind a bit of that. The reality is morbidity is expensive. Though if we keep on making progress, eventually humanity will have the means to make ill health relating to aging optional, and so that's fantastic. While at first, unfortunately, anti-aging treatments might be very expensive, however, as our understanding of anti-aging science increases, our engineering techniques improve, and with mass productions we achieve economies of scale, then medicine and therapies will become cheaper. At some point, we'll have the means to create affordable therapies for everyone, which will have an enormous humanitarian. Like all other medicine, the production costs of development will ultimately drop. We have no reason to think that this can't be the case for regenerative medicine. If the therapies remain prohibitively expensive, such that only the rich can afford them, it won't be because the real costs of the development of the therapies remain high. It will be because inflated profit margins are put on top of the real costs of production. Failing to achieve affordable rejuvenation therapies sooner rather than later won't be because it's impossible. It will be because we fail to coordinate and act sooner rather than later. And this goes for the development of the therapies, as well as working out the social implications of having the therapies around. True anti-aging is on the horizon of possibility. There are colossal implications for achieving this. Yes, indeed, it's going to change everything. But technology always
Yes, hello. I think with the end of this video, I can start. Yeah, thanks a lot for giving me the opportunity to share this last session, and congratulations to you all. We're hitting the home stretch and we are still around 100 people. That's awesome. Yeah, my name is Alexander. I'm on the board of HEALS and also founded the German HEALS. And now in this last session, we have the small task to solve all the legal, scientific, technical, and political aspects concerning clinical tests and biomarkers. And I directly get to our speaker and co-founder, president of HEALS, Didier.
Didier, you're mute.
Didier, we can't hear you.
Thank you, David. So this explains why you, okay. So thank you, hello. So this is about how to favor clinical trials for longevity. And first I will begin with a few facts and scientific aspects about longevity that most of you know, but probably not all people who will take a look at this video and at this conference. So the first one is today, like every day, about 110,000 people will die of diseases related to old age. And this is the first cause of mortality and morbidity in the world. It's about 70% of deaths in the world. It's about 90% of deaths in a country like my country, Belgium, or like in the United States. But even in the poorest countries, it is now more than 50% of the causes of death, diseases related to old age. We know that people are dying from cardiovascular diseases, from cancer, from neurodegenerative diseases, but we forget that is the elephant in the room, that even when dying of infectious diseases, even if when dying of falls, in most cases, the frailty due to age is related to this. And of course, concerning COVID-19, it is also this situation. We also know, so more the people here than other people, that the process of aging is something incredibly complicated. So John further is making this beautiful schema each year to explain all things related to aging and how we can understand all processes and what's happening, but also there are many things that we still don't understand. And there is one important aspect for me that it's, I would say, too often forgotten by longevists. It's the fact that at the moment we still don't have any way to go up the maximal lifespan. We really don't progress a lot. So we had a gain of 20 years in the last 2000 years because people were already living until 100 years 2000 years ago. The oldest person ever, Jan Calment, was probably 120 years, and the oldest person in the world at the moment is only 117, and the oldest man at the moment only 111. I think, and I think that many of you think, that it's very probably possible to find a treatment against aging within 15 to 30 years, but it will be complicated, expensive, and also we need for this, we need clinical, among other things, good clinical trials. So what do we need for clinical trials for longevity? I think one of the major aspects is simplification. So we have more scientists than ever in the history of humanity, but we have also more bureaucracy than ever in the history of humanity. One of the consequences is that, for example, it's costing about 1 billion to test for a new drug in Europe or in the United States. I even read not so long ago that it was now about 2 billions. It is primarily due to very heavy, very complicated regulation, and going very slow is not at all a guarantee to have things going better. So we need less bureaucratic and faster system of authorization at the European level, at the American level, or even maybe we can dream about a world level ever. At the moment, an average IRB, institutional review, so the ethical committees in the US takes many months, and the preparation of this, asking for this takes even more time. And an authorization is mostly only valid in one place, and certainly never valid in in more than one country. Even in Europe, who is supposed to be more or less a unified place, when you have an authorization in one country, it's not valid to another. We need kind of a paradigm shift. We need a moonshot, a Manhattan project for clinical trials. And at the moment, what's happening concerning real test for longevity? Well, not much. There is the TAME clinical trial, but still not really started. Normally, the international classification of diseases is supposed to better include disease related to old age, but this will not be, in my opinion, enough. For clinical tests, we need, of course, double-blind experiments, double-blind trials, one group with the best already available treatment, and one group or a few groups with the best already available treatment, plus the new therapy or the new therapies. It's important to remind that for these people, even if they don't have the new therapy, they will be in a better situation than ordinary people, because the following and the fact that they will be better followed will be better for them. We need volunteers, but at the moment, what we don't have for clinical trials is people who are old enough. So 70, 80, 95 for men, 99 even for women. Well informed enough, of course, in good health. Well, in good health, that's relative, and interested to work for themselves and for the community. What we also need is good products or good good therapies. So this list you probably know already. So of course, gene therapy is not yet there. There are also aspects that we don't know. And so this was my list just until a few, let's say, months ago, but now I would add blood therapies. This is, we spoke about this at the beginning of this meeting. This is really very promising, and I hope and I heard that clinical tests, especially with old people, will begin soon. We need, and this is also what was approached, especially today, we need good biomarkers and public results before the treatment, during the treatment, and after the treatment. And of course, especially when the results are not what expected. That is one of the big problems. You all know that probably, that sometimes there are clinical trials or other experiments who are made, and you never see the results because the results were not what was expected. Below you see one of the non-completed list of possible biomarkers. Okay, so we need, like I said already, a global project, a kind of Manhattan project. Sometimes it already happened in the history of the humanity that there was kind of a decision to change things, like the first man on the moon. There was also the war on the cancer, who was less successful, but still it was kind of a successful story for many aspects. We need, of course, money. In my opinion, it should be more public money than private money, but this is for another time and for another discussion probably than this one. We should, of course, use more artificial intelligence. That was already approached today. And what I wish is no patents. That's my wish. That's not what we maybe absolutely need, or at least not for everything that it's paid with public money. Because there is a risk that it's not usable for everybody, and also because there is a risk that it's going to slow down research concerning aging. So I'm already going to the conclusion, and I hope after time for one or two questions. So we need this to have more equality and more equity tomorrow. We need this because it's our duty as scientists, as social activists, to rescue people. And thank you for the people from HEALS, from the International Longevity Alliance, from the Life Extension Advocacy Foundation. These are organizations among the, well, many and increasing number of organizations working resolutely in these directions. Thank you.
Thanks a lot, Didier. There's actually one question from David Wood, but I would propose, because of the time, David Wood first gives his talk and you have the time to answer the question in the chat.
Okay.
Alexander, shall I start?
Yes, please, David. Sorry, David Wood, who doesn't need an introduction. Please start.
That's fine. So I'm going to talk about the possible surge of public interest in biomarkers of aging. So that if we come back to this conference in 4 or 6 years' time, we won't be having the same old discussion. Oh, we wish there could be a moonshot project, already there will be a change in public attitude. I'm going to take a little bit of a futurist attitude in here. And by futurist, I don't just mean predicting something that might happen in the future, looking ahead to a desirable end goal that I guess all of us in this call probably share, that there will be at some stage comprehensive, effective longevity therapies, inexpensive, in wide use. And we might imagine optimistically 2040, pessimistically 2140. No, I'm not just going to talk about that. I'm going to use the technique of backcasting. That's a rather grand name for a simple idea, which is that we must look in the intermediate timescale. What kinds of things do we need to consider for the short term that will increase the likelihood of that desirable end future happening sooner rather than later? One possible intermediate goal, of course, is that there is a lot more funds invested in this area, private money and public money, but we have to backcast again. What's going to make that increase in funding possible? And that takes me to the possible influence of celebrities, or we might call them key influencers. So let me just ask you to indulge me for a minute in a bit of fantasy if we look ahead 5 or 6 years. People like, I wonder if you recognize this group here. This is the Beckham family. David Beckham is a famous soccer player or football player. His wife, Victoria Beckham, used to be known as Posh Spice. Where's this going? Bear with me. The Spice Girls, for those of you old enough, you might remember them as, in some ways, the most popular and successful all-girls group ever. They still have more records sold than any other such group. What's happened to Victoria Beckham? Well, she now has a beauty business, and on that beauty page today, you can buy something, it's quite expensive, a cell rejuvenating power serum, which is claimed or suggested isn't just a cosmetic. In some ways, it actually rejuvenates the skin. And if you click on that, you will find various claims about chemicals that sort of might have been mentioned at our conference today. Niacinamide does such and such, fragmented hyaluronic acid does such and such. Well, does this really rejuvenate the skin? Hold that question. Another possible celebrity, you may not know this person. He is a professional golfer. He is 27 years old. His name is Bryson DeChambeau. I am picking him in part because he has recently done something that only 2 other professional golfers have done. Even if you don't know much about golf, you might know one of these. 2 people who have done the same thing. Tiger Woods dominated professional golf for much of the last 20 years, and the Golden Bear, Jack Nicklaus, was sort of in the same role for the previous 20 years. And all 3 of them have uniquely won the 3 particular championships: the US University Golf Championship, the US Amateur, and most recently the US Open. Why am I telling you about Bryson DeChambeau? Well, his fellow golf professionals nickname him the Professor. He applies lots of mathematics and physics and biology in his approach to golf. He has made all his golf clubs the same length, defying conventional wisdom. He talks about mathematics a lot, and he has created his own diet that in the last few months, actually in this year, has put on 20 pounds worth of muscle, which means he now hits the ball incredibly hard and incredibly fast. Why am I telling you this? Well, he has explained his diet in an interview in GQ magazine recently. My goal, he says, is to live to 130 or 140. I think that's possible now. Well, I think he's wrong there, of course. But he's more plausible when he goes on to say, I think that somebody's going to do it in the next 30 or 40 years. I want humans to be better. I want them to succeed, he says. And the rest of golfers are divided about this new approach. Some of them say, it's not golf, in the way that we British might say, it's not cricket. Others are more humanitarian minded. They wonder, is this really good for his health? Beefing up his muscles like this, might it actually foreshorten? And that brings me to the main point. We have to backcast again. Before the public as a whole can evaluate credibly what claims people like Posh Spice, Victoria Beckham, or Bryson DeChambeau, people with a great deal of influence and clout, what they may be saying, we need to raise public interest in credible biomarkers of aging, things that can be measured and can tell us whether these therapies are working. So I'm envisioning in a number of years that when people meet together, they won't just talk about current statistics. They will be sharing, hey, here's my biomarkers of aging. Oh, here's my biomarkers of aging. Or you should see my girlfriend's biomarkers of aging. Wow, really indeed. And bear in mind, we humans do like to exchange information about statistics, whether it's sports statistics, golf, professional football, ice hockey, whatever, music statistics. Increasingly, we are aware that our financial creditworthiness influences a lot of what we can do and not do. Some of us still talk about horoscopes or modern versions of that, personality types, preferred learning styles, sexual orientation, and yes, there are some discussion of health statistics, such as blood pressure, cholesterol. But I'm here to raise the possibilities that we will be talking in a few years' time. And when I say we, I mean the general public, or more parts of the general public, about their effective biological age. And I agree, by the way, with Walter Crompton, who raised the possibility in chat that we might need more than one such metric. So what would the implications be if that were to happen? As Alexander Zavoronkov already suggested, it could have implications in insurance premiums. More and more people might hear, if you want to lower insurance, well, today you can get a lower insurance in some cases as a car driver if you agree to put some new technology in your car that monitors your driving. In the future, you might get a lower insurance premium if you can prove that your effective biological age is younger than the norm. We might use it to evaluate potential long-term romantic partners. Companies might use it to evaluate employees ahead of giving them a significant new responsibility. And critically, we may start changing our own behavior or influencing those who we love to change their behavior because we know what our effective biological age is, and we can already see what changes we can take part with in order to reduce it. Is this all upsides? Well, as a futurist, I want to counsel everybody to think about the downsides as well. What could go wrong if there is, for a while at least, some widespread interest in effective biological age? We might anticipate some opposition to it. We might say people saying, don't discriminate against us, please, just because our effective biological age is older. I'm not so worried about that. I think we could win that argument. It's different from ageism. Ageism is when people are discriminated against just because of their chronological age. There might be more sense in discriminating against people who have large, inflated, effective biological ages. Then there might be disillusionment, a hostile backlash, first of all against therapies, which don't deliver the changes in effective biological age that they promised. I'm not too worried about that either. It's actually good if we can catch out therapies for not doing what they promise. It's good if we know that quickly. The bigger backlash, and a more dangerous one, might be if the biomarkers turn out to be bad predictors of mortality, not just in a few individual cases, but statistically as well. So we do need to do what Sven has already been arguing. We need to evaluate the various biomarkers from a number of criteria. And in addition to the criteria he listed, I would want to list these socially normative criteria too. And very briefly, to look at how innovations spread throughout society. Here I'm drawing on quite old work by Geoffrey Moore, but I think this is still very relevant. People in society vary in how well we are interested in crazy new ideas such as biomarkers of aging. There is an initial group of which almost all of us in this call belong. We care about things because, hey, they're cool, they're neat ideas, and they might, if we work hard, be helpful for us. What Geoffrey Moore points out is there's a big gulf, a chasm, before more ordinary people, real people, or mainstream people will adopt these new ideas. They want something that's simple to use. They want something that's got good usability. They want something that many of their colleagues, their social friends, their celebrity that they like, that they are already using. People who in some sense they feel are like them. And if we can get it right, Geoffrey Moore argues we must find a few killer apps, something that will appeal to some of these people in the majority, and then it can cascade with more people like them seeing that their friends are talking about biomarkers of aging, and they will start getting involved too. Thus, if we get it wrong and there's a backlash, there can be a cascade of negativity as well. Which takes me to my conclusion. I think it would be great if we can enable a surge of public interest in biomarkers of aging, but to do that, we need to be sure that we can communicate them well, including their drawbacks. They must be sustainable in the sense that we're not going to change our minds about them, and we must avoid the adverse publicity. And to make them successful, we tie them with applications with real-world relevance, known as killer applications, perhaps insurance. Perhaps, as Aleksey Moskolov mentioned, there might be some indication to your likelihood of being vulnerable to a COVID infection. Or, as I've seen on Twitter, a suggestion by David Doherty, they could be relevant in other animals, like the field of horse racing, in which huge money is invested. And if people find out that biomarkers of aging could be applicable to the treatments given to horses, then it could surge there well and then be copied back into human treatments. So insurance, COVID, horse racing, and possibly others. And if we can do that, then we may well inspire these key influencers who are not at this conference today, but who might talk to their very large followers on Instagram, Twitter, and elsewhere about the importance of this. And we can therefore change the public landscape with more and more funds then being directed profitably to this area. Thank you very much.
Thank you, David, for this, for your inspiring talk, and also for keeping the time. There are actually no questions, but a lot of answers from Didier, so you might take a look in the chat. And now we come to our last speaker before the general discussion. So Keith Comito from the Transhumanist Party.
All right, can you see my slides?
Yes.
Yep. All right, so as introduced, I'm Keith Comito, president of the nonprofit Life Extension Advocacy Foundation, actually, or Lifespan.io, which raises funds and awareness for life extension research. I believe most of you know who I am, so I'll proceed. Firstly, thank you for inviting me to speak at this excellent event. I'm happy to be here, especially today, the UN International Day of Older Persons, a perfect occasion to reflect on how addressing aging will serve to protect the older among us, especially in these times and everyone in general. In this presentation, I'm going to talk about some issues inhibiting biomarkers of aging achieving large-scale utility and adoption, how we might take some lessons from other fields to overcome them and potentially create a path to selling the public, investors, and policymakers on the value of aging research more generally and hasten the development of the field. So in that case, the latter parts of my talk, David actually teed up wonderfully. So when we get there, you'll see. But firstly, to begin, as we all know, technology has been rapidly developing that is allowing us to generate and collect various forms of biomarker data, even passively as we go about our daily lives through devices like mobile phones, sleep and activity trackers, etc. And this data is already being used in various clinical trials, and along with other types of medical data, is starting to be integrated into emerging healthcare data commons platforms, such as the NIH's All of Us initiative and the UK Biobank. And this naturally begs the question of whether a reliable system of aging biomarkers can be applied to data sets like this and become part of basically every trial to always have that secondary indication of how an intervention is affecting the aging process. And over the past few years, there has been a large number of age-focused healthcare products and online data systems emerging, aiming to use various biomarkers to illustrate their efficacy. But one of the main problems thus far has been that everyone's doing their own thing, using different data sets of biomarkers for testing similar interventions and or formatting their data in a proprietary way that doesn't interoperate with other systems. And this siloing of data, fragmentation of analysis methods, and lack of clarity on which biomarkers to use how and in what combinations creates a scenario that slows progress in comparison to one where everyone is operating within a standardized, goal-oriented framework. And this state of affairs indicates that we might be able to apply a lesson from the history of another field. With what we might actually need right now being a grand standardization initiative with authority and funding at the national or international level, like Didier was just speaking about, similar to what happened with the birth of the internet after the creation of the ARPANET in the 70s. This basically took the form of international councils set up to agree upon a set of protocols and standards so that everyone's work could interoperate. And this resulted in the backbone for protocols like TCP and HTTP, which are still used today and part of the infrastructure that's allowing us to communicate here right now. And importantly, analogous standards for health data like FHIR, or Fast Healthcare Interoperability Resource, are emerging now and starting to be integrated into ubiquitous technologies like smartphones, part of Apple's Health Kit, for example. And thus, now is the time to perhaps push for a similar grand initiative formed to select biomarker targets and interoperability standards so that the entire field can work together as we're working on our individual projects, studies, and companies, etc. Now, this is usually the part where statements like biology is not the same as software are made, and that is totally valid. And this is why it's important to note that the standardization we're talking about here doesn't mean standardization of the tests themselves, but rather of the format of the data that results from these tests, as well as in the process of how analysis and meta-analysis is conducted across various data sets. For example, simplified, let's say this hypothetical council is first able to agree upon some desired metric for biological age that, if magically achieved, would be unequivocally useful as a diagnostic to detect rate of aging throughout any clinical trial, specifically aging focused or otherwise. Say, for instance, a property of this is that it will be testable by being able to predict the chronological age of a normal person not undergoing any specific intervention, with a mean absolute error of less than X, with the X years, with a correlation above Y, etc. Whatever that is, whatever is deemed to be universally useful. Then, once that is determined, the requirements of how this goal could be achieved can be worked backwards from. Which types of biomarkers, with which properties we think cover each other's weaknesses, that we think are minimally necessary so that a composite metric based upon them will fulfill the desired properties of the goal metric. And it's important to note that this can be mathed out to some degree. Whether every biomarker potentially contributing to this is actually real or implemented yet or not. And then we can further work backwards to determine subcomponents in a similar process, creating a network of interrelations that I think Aubrey was talking about earlier. Solutions can then be analyzed by the council and debated upon with respect to considerations like Aubrey talked about, overfitting, etc., to determine which paths are the most feasible in the near term with the fewest amount of currently unrealized holes in biomarkers, what those holes are, creating essentially a scaffold that allows fulfillment of the true initial goal to be distributed and worked on by everybody, component-wise. Because we're all building individual projects to fit into the same overall spec and generating interoperable data while we're doing whatever else we're doing. Furthermore, because it is the goal that we're standardizing to, not the exact way in which we get there, this allows for iteration and flexibility regarding which tests and analysis methods are appropriate for which circumstances. For example, if we have a trial where we're collecting stool samples and blood samples, this combination of 10 will eventually decide would be the best route to the goal. Or what if we're doing a crowdsourced trial and all we can get is physiological biomarker data? What configuration of those markers and analysis methods to use in that scenario would fulfill the goal to the best of that ability. Now, if this seems a bit abstract or unrealistic, allow me to illustrate an analogous example involving data from technology you likely use all the time, video streaming platforms. With such systems, it's common for a company to want to predict things like a user's likelihood of staying with the service year after year, and to determine the most useful combination of contributing metrics that can be measured right now that accurately represent this future behavior. Furthermore, this process can be iterated upon, as with each year you can check it. The accuracy of various approaches and combinations can be validated to see which ones, which markers, and what combinations led to the most accurate predictions, and thus identify the most impactful targets for intervention. For example, let's say a higher number of sub-profiles on an account that also uses party mode frequently greatly contributes to user retention, then it would be smart for the service to work on interventions that promote use of party mode amongst multiple profiles on an account if they want more user retention. And now, it's important to note that this isn't really just an analogy. Some of these measures are actually biomarkers, such as decision-making speed. And this is gathered typically longitudinally, sometimes on a daily basis, from millions of people. So this ties into the second main point I want to make, and which David was alluding to, the opportunity to use physiological biomarkers as a key of sorts to convince and excite the public regarding the value of biomarkers in aging research generally. As mentioned, ubiquitous devices like smartphones are already passing or passively collecting a variety of biomarker data, and many have built-in diagnostics and frameworks like the research kit we mentioned, which facilitate the usage of this data in clinical trials. And online, people frequently submit data, like with FaceApp, that could be repurposed towards relevant ends, such as training facial biomarkers of aging. And this sort of physiological biomarker data will only become more regularly collected on a massive scale as technologies like voice recognition, augmented reality, and gesture tracking become part of those ubiquitous platforms, such as the streaming services, visual reaction time, tremor detection, etc. And this allows opportunities to really do something significant with just physiological data that can not only be useful, but also build significant public pressure for age-related biomarkers. As an example, what if it turned out that a marker related to aging, such as slowed reaction time, was an early symptom of COVID-19, which could be detected by the aforementioned platforms at a population level, a large percentage of people in a certain city, for example? You'd have potentially an early warning system. And you could imagine the headlines, your Netflix queue could save your life, etc. And how this could naturally build demand for data to be used this way and facilitate crowdsourced clinical trials, creating natural collaboration paths between the public, government, and large recognizable companies that can serve as powerful social signals. Now, of course, as mentioned, to do this right, there are related issues regarding data security and privacy that would need to be sufficiently addressed to make everyone feel comfortable. And they can be, and certain countries are already implementing interesting ways to do this with technologies such as blockchain, et cetera, but that's a separate topic. And to return to that hypothetical example, it also illustrates another knock-on benefit of creating the scaffold that we discussed earlier. It allows non-obvious stakeholders to see how they can be involved once such an initiative is created. For example, certain companies in the entertainment industry already have capabilities to gather specific types of physiological biomarker that surpass those of any current biotech company. And this would allow them to say, hey, that component, we can do that, and receive a related grant or inducement prize from the Grand Initiatives funding pool allocated to this, and or the PR benefits of helping to save lives. And this power to engage a variety of stakeholders is why I strongly feel having a grand government-led initiative to do something like this is a key plank of fulfilling the promise of the longevity dividend, which we spoke about earlier, and a topic which I spoke about more fully at the Ending Age-Related Diseases conference a few months ago, and you can find that video on our YouTube if you're interested. But in conclusion, a potential strategy to drive the development of aging biomarkers quickly may be to apply a similar data standardization approach that helped birth the internet ourselves as a community with physiological biomarkers, and think of ways to partner with high-profile entities, like David was talking about, to positively shock the public on the value of biomarkers generally, which can allow us to build momentum towards a large, grand initiative, which can in turn help us do this for all aging biomarkers, driving forward our field and enabling the potential for new types of crowdsourced clinical trials, which can not only allow more stakeholders to explore therapies lacking traditional profit motives, but also create pressure on the existing system to evolve. In this way, employing a model of standardization and interoperability for physiological biomarkers, similar to those that are already being employed on analogous data sets, can serve as a key to unlocking the longevity dividend and help propel us forward to a world free from the diseases of aging. Thank you for your time and attention. And I believe everyone here is familiar with what we do at lifespan.io, but if you'd like more information or to help us, here's that information, and you can contact me as well. Thank you.
Thanks, Keith. Also being so very punctual. So there are two questions. Have you considered, one's from Anton Kulaga, have you considered about standardization of benchmarks for measuring accuracy and other metrics of biomarkers, as well as distributed data set for cross-validation biomarkers predicting models?
Yes, absolutely. The case that I'm walking through here is kind of a simplified case. In that slide that kind of had the network of everything talking to each other, you would want to have systems like that to basically cross-reference all the biomarkers with each other in interrelationships. And basically, the key there is that guideline or standardization system isn't really just one monolithic thing. That's not what the council would decide upon. It's really a hypothesized system of mathematical equations that sort of relate them in relationships that then can be tested. So that's why I mentioned the point of, at the end, this isn't really what we want as per the earlier conversation, but it should be a property of a marker suite like this, that it should be able to accurately predict the chronological age of a non-intervened person, right? So that's not really what you're going for the interventions, but it should be a validator. And then you can see and iterate on the system. Okay, this configuration with these biomarkers seem to be more accurate than those. This configuration of biomarkers, the same configuration with different data sets, this data set seemed to work better than those. Is that because there's something wrong in our system, or because there was something lacking in that data set, et cetera? So it's a very complicated mathematical mesh, but my point is, we should set up a scaffold so that we have a place to start and iterate. And that's a process that I can't speak, without getting in trouble, but it's a process that I participate in to some degree in my other life. And I know that it's possible for very analogous data sets.
Okay, thanks a lot. And one short last question from Attila. Which existing organization do you think is closest to implement your approach?
To be honest, I haven't really done like a full audit of everyone who's working in the space, because as mentioned, there are a lot, especially in the last couple of years, which is a good sign. But I will say, to call out some of the earlier talks, Alex Zabranka, for example, was illustrating sort of a case of this, of relating different biomarkers to each other, testing them, putting them in the suite, developing, an aggregate clock. So certainly that type of work is moving in this direction.
Thanks a lot, Keith. And then we come to our general discussion with some help from Isabel, George, Jose, and Rajan. Please unmute yourself. And whoever feels free to go first, please just start.
I defer to Isabel.
I should start? Okay. Hi.
You're listed here first.
I'm listed first. Okay. Yeah, I don't really know what to say, but I read in the email that we should somehow conclude what we learned today and what was our favorite. So my name is Isabel. I have a PhD in genetics of aging, and I'm the scientific spokesperson of the Forever Healthy Foundation. And I really enjoyed today's meeting. I think also the structure was really cool, how we really went from the clinical testing to defining biomarkers to also now defining what we need to have it more accepted in the public, and also more the ethics behind it. I think what we learn is that there will never be this one biomarker we heard about. We can have transcriptomics, epigenetics, or proteins, even proteomics. They need to be sensitive and robust, and they need to have clinical relevance. And I think what we learned today, what I also learned today, is that AI is a big help in this. So for myself, I have to say I learned a lot. Omics is something that is coming, and I think in combination with AI, yeah, this will be really helpful in developing new biomarkers, but also we already have a lot, like the frailty index. I think it's always a combination. Yeah, it's definitely crucial for the aging field. I used to work with C. elegans. They just died after 30 days, so it was quite easy. For humans, we need to have biomarkers. But I was really positive today because I feel the field is really moving forward. And with that, I would like to give to George.
Okay, thank you,
Very
Isabel.
positive remarks, George.
Okay, so I study more disease of degeneration than basic aging itself, and I learned a lot from this meeting. One of the things is just highlighting that in most of the developed world, what people are dying of is degenerative diseases of aging. In fact, the estimate that 90% of the people dying in the United States are dying of degenerative diseases. It's not a surprise to me, and even infectious diseases impinge on it. But modern medicine has very little effectiveness in any of these diseases. We're only hitting them at the periphery and barely there. And I think this is because there's a lack of concepts. Medicine has been so long in dealing with diseases that are from the outside, predominantly infectious disease or diseases of obvious defect. We don't know how to deal with diseases that are part of our normal biology, and aging is part of our normal biology. And I think the type of approach used here, which is you need new concepts, but it's hard to get to new concepts when you don't even have an idea what concept to fill it with, and you need data. And the biomarker approach is definitely one of those ways to get at that, especially when it's used in an a critical, unbiased manner, either using AI, biomarkers, nucleic acids, proteomics. All of those can yield substantiation for things that have been studied before, but never were studied so eloquently. I was very impressed with the work of showing glycoaging, because I know years ago we found major glyco changes during Alzheimer's disease, which play a role in polymer formation of tangles and plaques. I was also impressed of the therapeutics that were presented. The alpha-ketoglutarate drug. Alpha-ketoglutarate is a therapeutic because in several neurodegenerative diseases, alpha-ketoglutarate dehydrogenase is one of the enzymes that's known to be altered during the disease very early, quite substantially reduced. There's an active site on that enzyme, which is very susceptible to oxidative stress. And I was also impressed by the frank discussion about the need to balance more scientists with less bureaucracy. If you just end up with more scientists and more money, and I see that in my field in Alzheimer's disease, if you don't have new concepts in an open field, you end up actually entrenching things so that it costs more money to develop drugs or actually impede progress rather than open it up. So I think scientists that have an open mind need to be involved in what processes need to go forward, balancing the ethics there. And then I know the discussion about lower insurance premiums for those that are lower risk. I would worry about the other contrary thing. With genomics and even further refinement, will people find that they are uninsured and should be dead? In other words, they have bad genes, bad health, etc., and we abandon them. So I think that the ethics part needs to be really discussed, and how governments and processes can balance those things. And the last thing, which I didn't see discussed, and I may have missed it, is when it's discussed that aging is this process that we're trying to modify. The fact that it occurs in essentially every organism, is this an evolutionary thing of a need to turn over us? Is it an organismal apoptosis and the process? It doesn't mean you can't modify it, but is it a process that if you interrupted, you would not have, you would have less fitness to the environment because you changed the turnover? And those are some of the comments I have.
Thanks a lot, George.
Jose? Okay. Well, greetings to everybody from Madrid, from Spain, in the middle of this crisis. And this is my weapon against COVID. We have to be prepared with our weapons. But first of all, I want to congratulate Didier and Sven for keeping this alive. The Hills Conference, the symposium, very important. I have participated in the 3 previous ones, and I enjoy them. And I think this is the biggest, at least in terms of virtual participation, so congratulations. And I also want to congratulate Ilya Stambler, because he is the one who pushed for the creation of the Longevity Day and then the Longevity Month. So this is very important, and I think that people need to be recognized. So Ilya, congratulations, and Didier and Sven for all your efforts. Basically, I have 4 comments. 4 different areas that I want to talk about quickly in order to push our goals for anti-aging and rejuvenation, and with metrics, because we need to measure things. I am an engineer from MIT, and I love to measure everything. So we need to measure, and in terms of social issues, I think David Wood, who is, by the way, my proud co-author in our book, The Death of Death, he gave some insightful ideas on how to get the public excited about this, and how we can get more and more information socially and acceptable. So I think this is very important. So that is the social issue. Now let me talk about the political issue, because Didier has also been involved politically in Belgium, and I have been also, I ran for the European Parliament. Actually, many of us here have been running for the European Parliament. Attila Sordas in England, he got 3,000 votes for the European Parliament. I got 7,000 votes in Spain from Madrid, 7,000 votes, which is pretty good in a quick campaign of 3 months for the European Parliament. And then Felix Wirth, he got 71,000 votes in Germany. And this is remarkable. Obviously, Germany is much bigger, and they have existed for 5 years, the German Party for Health Research. But taking these ideas into politics is fundamental. And my master idea for Spain was the creation of the European Agency for Anti-Aging. European Agency for Anti-Aging. And we wanted to copy, in a way, the US National Institute on Aging, but put a lot of data, a lot of information, so all these biomarkers and all of these epigenetic clocks would be fundamental for that. So after talking about politics and social, let me talk about technology, because we live in the most amazing time in human history, and technology is changing exponentially. I like to say that in the next 20 years, we are going to see more changes than in the last 2,000 years. This is exponential change, and the idea of the singularity with my friend Ray Kurzweil, where we work at Singularity University 10 years ago. Anyway, so what has happened is remarkable in terms of our field, absolutely remarkable. First, senolytics, CRISPR, the TAME trials, all the biomarking, epigenetic clocks, the Yamanaka factors, telomerase treatments, it's incredible. Nothing of this existed 10 years ago. So this is moving very fast. Technology keeps on advancing. And so I am very excited that we are going in the right trend. I would like it to be even faster, but I am not terribly disappointed, because I think we are getting to a tipping point, Also, we can see that in conferences. Today, I have been in 3 longevity conferences, 3. In the morning, the Longevity Investors Conference from Zurich. At midday, the Ibero-American Conference, Longevity Conference in Spanish and Portuguese, Latin America, and Spain. And finally, this other conference. So 3 conferences in a day is absolutely remarkable. And this year, there have been about, I don't know, but at least between 10, 15 longevity conferences. So this is becoming very popular. And so I get to the final point about this popularity, which is economics. And this is important, economics. And then obviously the longevity dividend initiative
also didn't really exist 10 years ago. And now, thanks to Keith and Liv, I think now there will be a relaunch with the longevity dividend version 2.0. And this is very important because people need to understand that this is actually economically intelligent. This is positive economically. This will create more revenue and less expenses if people are healthy, if people are youthful. So I think, just to recap, we are going in the right trend, socially, politically, technologically, and even economically. So I want to be positive, and I am pretty sure in the next European Health Symposium, we are going to have better ideas and better results. So thank you very much.
Thank you, Jose. Energetic as always. So please now the final comments or even slides from Ranjan.
Yes, hello. Good afternoon and night, everyone. Thank you so much for having me here. Very pleased. I'm probably new to every single person here, and I'm very thrilled to be part of this community today. Thank you to the organizers as well. It's amazing to see that so many people are touching on many of the same numerous themes today. It really shows sort of a critical mass building here towards a highly coordinated effort. Just a quick introduction, since I'm relatively unknown to everybody. I initially was involved with a nonprofit and with the Personal Genome Project at Harvard Medical School. That's where I cut my teeth, mostly in community building, in open source science, participant-focused science in the world of genomics. An unrelated project, which I'm currently involved with right now, is called RADVAC, or Rapid Deployment Vaccine Collaborative. That project was motivated by the obvious harm occurring right now in the COVID-19 pandemic, and in the recognition that current structures in place for vaccine development and pandemic response have significant shortcomings, as all highly well-defined systems do. And it's based primarily on self-experimentation. So this open source IP-free platform was primarily developed by Preston Estep, genomicist from Harvard, who released this formula for a vaccine platform to accelerate vaccine development around the world. And it is entirely self-experimentation based. So a number of us.
Ranjan, to interrupt, are you actually showing slides? Because we can't.
I am not. I apologize. I should have clarified.
Perfect. Then that's great. Just to make sure. Sorry for interrupting. Yeah.
Okay. And I apologize. In fact, my name, I am logged in twice, so I see that my image is not coming up. Why don't I just? I'll just go ahead and switch my audio here.
Just continue. We highlighted your video. We spotlighted your video.
All right. So this project is based on self-experimentation. So a number of us have actually experimented on ourselves with this new vaccine formulation, and are currently testing samples and validating data and developing new testing methods. So one of the things that Didier asked me to address was this theme of self-experimentation, of open source science, of individual level science, citizen science, in the context of what is being discussed today. And this, of course, varies by jurisdiction, but these sorts of things obviously depend highly on a well-informed public and citizen scientists who may not be professional scientists, although in some cases they are professional scientists working outside the boundaries of institutions, but citizen scientists who are interested and willing and educated enough to carry out some of this experimentation on themselves. And that, of course, relates to what was discussed here. Josh Middledorf's talk, in particular, on the observational trial points to the fact that a large number of people are using interventions on themselves. And so there is significant value in following them, collecting that data and maintaining connections with them. It's also about accessibility. In light of the vaccine effort, we know that with the vaccine situation, higher income countries are going to have access almost immediately to the vaccines when they're approved. They've been pre-manufactured, of course, in some cases, whereas lower income countries will have to wait 6 months or a year or longer. And so this was motivated also by creating an independent effort that was outside the bounds of the conventional way of doing things to bypass this delay in saving lives or preventing morbidities in light of the vaccine crisis, which I would say also applies quite a bit to longevity technologies. The landscape is completely changing, of course. There's a new world, one in which technologies are more accessible, both through the lower cost, as well as miniaturized equipment, accessible supply chains, and customizable manufacturing capabilities, desktop manufacturing, things like that, and the wide availability of information. So the average person, average citizen scientist, can access almost many things and more as paywalls come down, and educate themselves and take matters into their own hands. So that's a very real landscape that we're going to be dealing with, and tying this into the legal framework and regulatory framework, those things are going to have to keep up because this new reality is a reality. Just a few words on citizen science. I agree with much of what was said. Citizen science is an incredibly powerful, untapped resource. Crowdsourcing was a word that was used today, which is very much related. By inviting participants in observational trials or in distributed research efforts to be co-investigators and bringing them into the conduct of certain studies, we can leverage this to enable easier iteration on research. So by maintaining, for example, contact with study participants who may not be in a traditional clinical trial network or structure, it's sort of a hybrid situation where people are feeding into, with their data into a distributed research effort and can be recontacted for additional testing and questionnaires. Oftentimes, the newer research sheds light on what older research has shown. And wouldn't it be nice not to have to execute an entirely new clinical trial to test a revised hypothesis, but simply to get in touch with participants of a past study and collect, incrementally more information or additional samples to update the hypothesis instead of outlaying tremendous amounts of time and money for a completely new investigation. So there's too much to discuss here in such a short time, but I'll leave it at that.
Thank you, Ranjan. So this being German, so I'm very happy that we saved all the time again, and now it's 10:15. So we take a very short break of 10 minutes to finalize the text or the declaration. Yeah, Jose kind of took already the words I wanted to say, but I just gonna say the names again. Really, Sven, Didier, Ilya, also Virginie, awesome what you did. I really enjoyed it. But nevertheless, I'm really looking very much forward to seeing you or meeting you all in person again, hopefully next year in Brussels. So bear with us, 10-minute break, and then we are there for the very, very final session.
Expensive such that only the rich can afford them, it won't be because the real costs of the development of the therapies remain high. It will be because inflated profit margins are put on top of the real costs of production. Failing to achieve affordable rejuvenation therapies sooner rather than later won't be because it's impossible. It will be because we fail to coordinate and act sooner rather than later. And this goes for the development of the therapies, as well as working out the social implications of having the therapies around. True anti-aging is on the horizon of possibility. There are colossal implications for achieving this. Yes, indeed, it's going to change everything. But technology always has. Life in the 21st century has always been one of constant change. We don't need to be paralyzed by fear. We need sober minds to address the issues such as overpopulation and therapy access, as well as the technical challenges involved in achieving anti-aging therapies. To ultimately solve the problem of aging, we need clarity in thinking about aging itself and practical approaches to mitigating the suffering it causes. If we don't rid ourselves of the diseases of old age, it will be our own fault, not an inescapable destiny. Biological aging isn't an unsolvable problem. We now have access to countless drugs and therapies to heal and cure ailments that once thought inevitable death sentences. We have solved difficult problems before, and now, more than ever, armed with modern biology theory and advanced tools to aid in science, this is the right time to focus our efforts on the right problems, which are often underfunded and under-resourced. To mitigate the risk of becoming complacent, we shouldn't confuse motivation with action. Let's ask ourselves, what are we going to do about it now?
I remember taking a walk in the woods with my dad a few years ago. We talk more now than we used to. We walked through pools of light and dark and talked about life. He told me how he thought it was neat that his father had lived long enough to be around horses and buggies, and then all the way to putting a man on the moon. We wondered where it was all going. As we walked and talked about times changing, I couldn't help to think that my dad was only still there with me in that moment due to technology. He had a heart attack in 2010. I remember getting the news in the cold and final week of January. He'd always had high blood pressure, and add to that a stressful job, and heart disease found him. They opened his chest, stopped his heart as a machine took over, took veins from his legs, and with them rerouted the blocked vessels to his heart, and brought him back. I thought about how miraculous that really was. The rapid advancements and dramatic increase in survival rates of heart attack victims is truly a hallmark of modern human technology. Only later did I realize that really what happened with my dad was a form of life extension. This could not have happened, say, 100 years ago, and now he was expected to live another 15 to 20 years longer than he would have. So really, life extension already exists today. Many of us may not think about the fact that some of our medications, surgeries, and better general health care are really forms of extension. With this kind of progress, it's only logical that we will be able to continue to extend human life even further, and perhaps indefinitely, perhaps even bringing back some youth. If we're already doing it to some degree and accepting it, and we think it's beautiful when someone makes it to 100, and we think it's tragic if someone dies in an accident or from cancer, I sometimes wonder why we accept death at all. I think it's all relative to what we're used to. Maybe in the not too distant future, it won't even be a second thought for someone to reach 150 or 200, and we'll look back on this age thinking it was weird to die so soon. Of course, there will always accidents, nature, and ourselves. But does that mean we shouldn't try? We are conscious and intelligent beings, and yet we have to watch as our own bodies decay and our loved ones fade. Here it is, 8 years later, and he's still doing well. It's weird to think if it weren't for this technology, the footage you're seeing now wouldn't even exist. I'm sure glad we have some form of life extension today, and hope that it grows so that us and future generations can say the same thing with even more time to share.
Okay, I hope this time I didn't forget to activate the sound. I hope you see me. And so this is the last part of this long conference. So this is the general discussion about the text that we were submitting to you at the beginning of the meeting, and we were recalling this quite a few times. The good news is there were a few changes who were proposed, and there was nothing, I would say, very sensitive. So I already agreed with everything. And so I give the link once again. And I propose to give the floor to first to all speakers and people who were chairing the meetings, if they want to say something about this text, or even if they want to say something in general, but then short. And after that, if there are people in the general audience who want to speak, they can also speak still about this text. And please shortly. Yeah. And okay, I want also to say, so this text is about how to significantly extend healthy lifespan with biomarkers and clinical tests. I really think concerning, well, we really think concerning clinical tests than one, but it's of course not the only one, but one very important aspect is how to accelerate them. And how to accelerate them is not going faster, is not going less prudently, sometimes in the contrary. And another part is how to have more open, more public and better biomarkers of aging. We have already quite a few websites where you have information, but there should be more coordinated action for this. So the floor is to the speakers and people who were sharing to want to have it. Please don't ask me, speak directly. Yeah, you turn.
I'll start. First of all, I want to say thank you very much, Didier. Thank you very much, Virginia, Sven. Was a really exciting event and very helpful if everybody admits. And hopefully it will be a basis that we can continue this discussion, continue to expand this consultation that will eventually have impact on decision makers. And about the text, looks okay to me. My only question is, I wonder where we should actually send it. Okay, publish on the website, fine, but we should also probably think whom we can address it, so it will have some additional you know, as an advocacy document.
Okay. Okay.
Yes, thank you for, I like the text for me, or thanks for accepting the modifications. I really like it. Yeah, I said thank you to everybody already, but really in these challenging times of COVID, I really like that we got so many people together for working on other really pressing matters for humanity. Thanks.
Okay. So I see, David, that you were writing normally a little bit too late, but okay, two things that I will share the screen to let see what you were saying and writing. So the can you normally you can see the chair so one is about the time limit is, and one other is about the fact that an authorization in one place should be valid for other place. For the first aspects, at least at the European level for me, or at the US level, for me, it doesn't mean that it has to be valid in the US and in Europe, but in the US together or in Europe together. So if this is enough for you, let us know. And okay, never more than 1 month. That could be going too far. Yeah, the first aspect is you can say never more than 1 month. Something like without justification, but you know, in my country, Belgium, you have this mechanism. And in other countries also, that you are obliged to decide in a certain period of time, and this is working. I mean, and this is working quite good, and this doesn't make it a less good legal system in general. So I think 1 month also for health questions should be, and it's also only to authorize or to refuse medical tests. I think this is enough. But I'm listening to you.
Not sure whether I'm speaking or not.
Yes, yes.
Yes, so on the point of variation, I think some diversity is important. That some of these questions as to what regulation should apply, of course, they shouldn't be unnecessarily different, but some of the questions as to what should be allowed and what shouldn't be allowed really are hard. And instead of insisting that every country adopt the changes at the same time, we should take advantage of some countries being more open-minded on some things than other countries. So that's why I'm just advocating a bit of support for diversity. In terms of timescales, yeah, we should put pressure on people not to take a long time unnecessarily. Again, though, I think some of these ethical decisions are hard, you know, as to what should be done. And if we're not careful, we might sound a bit like the people who want to rush through dangerous changes without proper consideration. Like there are risks with the new vaccines that are coming out, you know, these vaccines for COVID. Of course, we want to see them in place quickly, but not too quickly. And there are dangers to vaccines being approved before they are properly ready and then causing a public backlash. You know, why are there some people who are anti-vaxxers? It's because some vaccinations do cause harm or have caused harm. Some of the first polio vaccines did give people polio and killed numbers of people. So that's why I don't want to appear just gung-ho. Oh, these stupid ethical committees, they're just slowing things down. They are doing an important job. So we should pressure on them without coming up with an arbitrary milestone of 1 month. Anyway, but I do need to go. I'm sorry. So I will leave you to finish this off. You've done a great job today, but I'm going to duck out of this discussion now.
Just David, to say it again, I mean, the idea was to say concerning the authorization that you have, you have to have one authorization in the US, one authorization in Europe, and not in each part of the US and each part of Europe. And I mean European Union, of course.
Well, I'm a fan of the European Union, but not everybody is. And maybe some countries like Switzerland may want to do something different. So
yeah, so Switzerland is not in the European Union.
That's right, but
yeah.
it is in Europe. So
yeah, so I'm European Union. Yeah, okay.
Anyway, I mean, these are nitpicking, but I think we must understand why people are nervous about a uniform system being imposed. There are some merits to diversity.
Yeah, sure.
And then when we see, oh, this is working well, other countries can adopt it. And other experiments will turn out to be mistakes. And but we won't know in advance which will succeed. It's like we don't know in advance which biomarkers will work. We don't know which in advance which therapies will work. In the same way, we don't always know in advance which regulatory innovations will turn out to be successes or mistakes. So we want to apply great thinking to this, not just a uniform thinking.
Okay.
I know you're trying to finish the document, and I'm sorry for not reading it
Yeah.
earlier, but I was paying attention to the talks earlier.
Yeah. It's, thank you for your opinion, David. And so maybe no, the word is to all other people who were speakers today. So you probably have an opinion about this, and well, those who are still here, of course. Nobody wants to say something?
We can, discuss it later. We don't have to, accept everything this moment. We can let people think a little bit and, come up with a consensus in a couple of days.
Frankly, I'm not so much in favor. I prefer to have some kind of, let's say, weaker document, but that we can, or weaker in my opinion, but that we can, let's say, finish today than to wait for it. So I would say, for example, I can put normally not more than 1 month. But still, okay, let's say I understand the point of view of David concerning the diversity of research, but for me, it's very clear that at least once again, at the US level or at the European level, there should be only one mechanism of authorization and for ethical committees. And well, in many aspects of the US laws and also in many aspects of the European laws, when you have an authorization in one country, you have authorization in one part of the European Union, of the United States, you have the authorization everywhere. So, okay.
It's also a point to make a discussion. Somebody agrees, somebody doesn't agree. It will be boring if everybody agrees on everything. Our point is to make people think about it, so it's not so.
Yeah. Okay. Yeah, Daria, I see that you are changing many things now. This is a little bit complicated because normally it was, I wrote quite a few times on the text, please give your proposals now. Okay. Okay, I don't know how to, or maybe, how can we do that? Maybe another way then is to to finalize in a few hours. I don't know. I don't have any idea. But
If I
But
may
we can
propose something, I would say maybe people can add for 1 more hour, and then Didier, Ilya, and Sven in the morning, you decide on the changes, and then you publish the official document.
let's talk tomorrow, you know, on the fresh head, as you say.
okay, yeah, okay. So Alexander, say it again. I think we can
I
agree on
would put a
this.
deadline now for,
Yeah.
let's say, for 1 more hour, people can comment, and then in the
Okay.
morning, you as organizers decide on the final acceptance of
Fine. Yes.
suggestions, and then you publish it.
Yes, but okay, and maybe this we will send an email, or we can decide at which time we will meet, and other people are welcome to come also, so that it's let's say a collective finalization for those who want. But or let's say so that it's the same time tomorrow 5 p.m. we have the last discussion, and then we approve. Yes, okay. But Daria, if you can hear us, can you say a few words?
Yeah, sure. Great text. Generally.
Okay. But that's because you are proposing many small change, and to be honest, it's complicated if I have to see everything. But okay.
Oh, you are just let's discuss because you did great job. So I'm just kind of trying to make it in accordance to the law, and then this is all.
Okay.
It's only a few corrections. Tomorrow morning you will.
Okay.
Find
okay.
it.
So
you know, this is George Perry.
Yes.
I have just a simple question. Where is this text located?
On Google Drive, I was.
Did you send access to it?
Yes, yes. I put the link in the chat quite a few times.
Okay. I didn't read things.
So yeah. Okay. So it was there at the beginning of the meeting, and Virginie put the link 2 or 3 times.
No,
and
I just don't read all the chats.
Okay, yeah, it's complicated sometimes, and all people have access, but cannot approve changes, but they can propose change. Okay.
So great, thank you. I would really propose just 1 hour now to comment, and then you decide tomorrow.
So yeah, so the proposal of Alexander plus me is so you can propose things until let's say until midnight Belgian time, and after that tomorrow at 5 p.m. Belgian time, so the same time as today for the beginning of the meeting, the people who were chairing will decide, but everybody can come, and it will be the same link as today for this final finalization of the text. But now maybe last thing is if there are people who were not speakers who want to speak now, especially about this text, is your last chance. Otherwise, we will have the end of the meeting, of the conference. Nobody?
No.
Okay. So I'm very happy that globally things worked well for this conference. I'm very sorry that I think Alex, no, who was not, one person was not recorded on Facebook during a few minutes, but we will try to find a solution. This should normally be available later on YouTube. I hope that there will be other conferences also trying to discuss let's say, specific aspects about aging, because there are many conferences, but maybe not enough specific, and also discussing text and ideas. I thank you once again for being here, so many people until so late or so early or after so many hours. And I now stop the recording. Yes.
Thanks everybody again.
Voila. So the I have to stop also on Facebook. Yeah. Okay. So